Just allow me a moment, sir. Yes, sir. So we are live. Shall I begin with my speech or? No, am I
No.
audible?
Yeah, if we are live, then we can start.
So you're not audible.
If we are live already, we can start.
Okay,
Right?
sir. Okay, sir.
So shall we
Yes,
start?
sir. We are live. Yes, sir.
Okay. Dr. Partho, please proceed.
Good afternoon, everyone, and a warm welcome to you all. I'm Dr. P.S. Datta, and it's my honor and privilege to serve as the moderator of this very special two-day virtual workshop organized by the Physician Research Foundation, West Bengal, under the Association of Physicians of India, West Bengal chapter. So we are joined today by some of the most eminent minds in the field of medicine and research. Over the next 2 days, we'll dive deep into the fundamentals of research methodology in medicine, a subject of growing importance for all physicians, researchers, and postgraduate medical students. So let us now begin the inaugural session. I would like to request our distinguished guests to deliver their welcome addresses. Dr. Jyotirmoy Pal sir, President, Association of Physicians of India. Dr. A. Muruganathan sir, Director, Physicians Research Foundation. Dr. Partho Sarkar sir, Chairman, API West Bengal. Dr. Uttam Pal sir, Chairman, PRF West Bengal. Dr. Ashish Kumar Shah, General Secretary, API West Bengal. Professor Nandini Chatterjee, Scientific Chairman, API West Bengal. And Dr. Shambhu Samrat Samadhita sir, Secretary, PRF West Bengal. Over to you, Dr. Jyotirmoy Pal sir.
Thank you. I congratulate PRF branch of Association of Physicians of India West Bengal chapter to initiate such time-honored session. There is a research methodology. Being a president of API, I have given due stress on the research. We 100 crore population India have, and we are many ahead in many of the fields: IT technology, space technology, pharmaceutical production, vaccinology, in many aspects. But really, we are not so much in the medical research. In this field, we are lacking. So, research should be the priority for the coming days physician. And to be a good researcher, you must know the good methodology. Unless your methodology is perfect, after doing a hard work for 2 to 3 years, maybe 5 years, 10 years, when you will produce your work in a reputed journal, it will be rejected because the methodology is not perfect. So that is why first duty of an organization is to train its members, students and doctors, physicians regarding the methodology. So I discussed with Dr. Shambhu Samarja, Dr. Partho, Dr. Uttam Pal, and definitely Dr. Muruganathan, our Director of Physician Research Foundation, API Central, regarding repeated course of just by doing a single course problem may not help. So repeated course of methodology may not be at central level, even in the college level, district level, will help to upgrade the knowledge and methodology and to start research in a perfect manner. So with this few words, I wish all success of today's program. And I hope all the, even this is done from the state branch, I hope the city branches also participate, and also they also start such methodology courses in their own districts. Thank you. Over to Dr. Shambhu.
Thank you, sir. Thank you very much. We are now really delighted to have with us Professor Uttam Pal, sir. Professor Uttam Pal is currently the Chairman of Physicians Research Foundation West Bengal branch. So may I invite Professor Uttam Pal? to kindly share his thoughts.
Yes. Dr. Sambo, am I audible? Yes.
Yes, sir. Yes, sir.
Am I audible? Yes. So good afternoon to all. Professor Jyotirmoy Pal, National President API, Dr. Sambo, Dr. Partho, and other faculties and participants of this session. Welcome you all to this workshop of scientific research methodology. This session will explore the systematic approaches and techniques used to conduct research in various fields. Various fields mentioned by the Professor Jyotirmoy Pal. There are various fields of research. Scientific research methodology is the backbone of any research project. We know all this. It provides the framework for designing the study, collecting the data, analyzing the data, and interpreting the result of the research study. This research methodology guides us through the process of research, from framing the research question to analysis and interpretation of the results. So, understanding this research methodology is crucial for producing a good research, good quality of research, which help us in advancement of knowledge. So again, I welcome you all to this academic session to gather knowledge and make these scientific sessions of research methodology a powerful tool and successful workshop. Thank you all. Thank you.
Yeah, thank you, sir. Thank you very much. Now we have with us. We will wait for other dignitaries, but now we have Professor Nandini Chatterjee, Madam, the Scientific Chairperson of Association of Physicians of India, West Bengal branch. So now may I invite Professor Nandini Chatterjee, Madam, for her welcome address.
Thank you, Dr. Samazdar. The Physicians Research Foundation wing of West Bengal API has been very enthusiastic in doing workshops which are of immense importance in the current scenario. Knowing about research methodology should be our priority as physicians, as students, as teachers. And this workshop, which will encompass every aspect of how to go about in doing this with eminent speakers who are experts in their field, I am sure will be very helpful for all our students and listeners alike. With all the yes to Shambo, Dr. Parthasarathy Dotto, the whole PRF wing, Professor Uttam Pal, and with assistance of ourselves, I just hope that we are able to do a good job and it is helpful for our students. Thank you.
Thank you very much, Madam. Thank you for your kind words. And now we have with us Professor Partho Sarkar. Professor Partho Sarkar is the chairman of Association of Physicians of India, West Bengal branch. So, sir, kindly give your welcome address.
Okay, good afternoon, everybody on the virtual platform. Actually, I am extremely privileged and happy that our Physician Research Foundation is taking this great endeavor on behalf of Physician Research Foundation, the scientific research methodology, which is an extreme necessity among the medical personnel, as because our country is such a populous country, very lack of different data about different diseases or different community-based data, medical data. So I think this program will help our junior faculty, our postgraduate trainees, and those doctors placed in the peripheral areas. They will be rejuvenated and they will be encouraged to do research, and so that in the research area we can generate some data which will be our own, the Western data. So it is of extreme importance. Previously, we had a GCB program that was a hugely successful program. And my thanks to the organizer and mainly Shambho to make that program so successful. I think this program will also be very successful. And I am privileged on behalf of API that PRA under the aegis of API is doing this program. So thank you everybody. So please enjoy the program and learn something. So over to Shambho again.
Thank you once again, sir, for your continuous support and guidance. And we will wait for other delegates also. But before that, as we are going to start our technical session from 3 p.m. onwards. So the thing is that it is a customary as the Secretary of Physician Research Foundation of West Bengal branch of Association of Physicians of India. May I invite you all? May I invite our respected chairpersons, honored faculty, dear colleagues and friends, warmed good evening or good afternoon and namaskar to you all. On behalf of Physicians Research Foundation West Bengal branch under the aegis of Association of Physicians of India, West Bengal branch, it is my pleasure and privilege to welcome you all to this enlightening session on understanding the research process in medicine. As doctors, though the name suggests, it dossier to teach, but we are lifelong students, constantly questioning, learning, and striving for the truth. The journey of research, in many ways, a journey towards deeper understanding, not just of disease, but of humanity itself. Allow me to share a spiritual reflection as usual from the ancient Upanishad, which reminds us, Tamaso ma jyotirgamaya, led from darkness to light. So in the world of medicine, research is our lamp. It dispels the darkness of uncertainty, illuminating the path toward better patient care, ethical practice, and scientific progress. Each thoughtful question, every careful study brings us closer to the light of knowledge and healing. Today's session, it is also tomorrow's, also we have a dedicated session. They are more than an academic exercise. It is a celebration of our shared commitment to rigor, ethics, and compassion. Whether you are a seasoned researcher or just beginning your research journey, you are part of a tradition that honors curiosity, evidence, and service. Let us approach this session with open minds and collaborative spirit, ready to learn from each other and to inspire one another. May our collective efforts today contribute not just to the advancement of medical science, but also to the well-being of our patients and the upliftment of our society. So once again, I welcome you all on behalf of the PRF West Bengal. May the session be fruitful, insightful, and source of a new beginnings to many of us who are starting the research journey. Thank you all. One small announcement will start from 3 p.m. But there are certain technical issues, if you are facing that, you please contact with us, and I request Clarnet team in your chat box, please share the number of your concerned person, and once again you please send email to all of the delegates who had registered. I think it is very important, because personally I got a few calls from different colleagues. They are having certain difficulties. I am really sorry for that, but I hope the team Clarnet will take care of these issues for our future endeavors. So thank you once again. I want to thank Dr. Partho Sarathy Datto for his continuous support and helps. So over to Dr. Partho Sarathy Datto.
Thank you, sir. Thank you, all of you, encouraging and insightful words. And your vision lays the foundation of this workshop's relevance and impact. So we'll start the next session from 3 p.m. sharp. Dr. Shamazda sir have already mentioned that. So we have to wait 10 to 12 minutes, and then we'll come again and start the first technical session of this workshop. Thank you. Thank you.
Hello.
Yes, sir. Good afternoon, sir. Sir, please, we want to hear from you, sir, for this workshop. So, sir, we are waiting for your inaugural speech, sir. So, and you are audible. So, over to you, sir.
Cannot hear, sir. Sir, you have to unmute. Muruganathan, sir, can you hear us? Hello, sir, can you hear us? Muruganathan, sir?
Yeah, please. Please. Good afternoon.
Good afternoon. Now we can hear you, sir. Yes, sir. So we are really delighted to have with us the Director of Physician Research Foundation, and he needs no introduction, Dr. A. Muruganathan. So may I invite Dr. A. Muruganathan for his blessings to this workshop.
Good afternoon, everyone. I'm really happy that the PRF wing of West Bengal State Chapter has taken the initiative of forming the research methodology workshop. I think every one of our physicians should be sensitized to see how even small things can be made into a project. Every day you come across so many search questions, research questions, there is no answer. So it is how we should form the research project based on our practice, based on our experience also. My dream is for every state, they should have at least 4 projects every year, which means we can cross 100 projects. It's not at all difficult to, start the research project. My request to all the members to coordinate with the medical colleges, because medical colleges has got a huge data. You can do retrospective study, prospective study, observational study, cohort study. So to know what is that study, what is cohort study, what is cross-sectional study, for that one should be knowledgeable about how to conduct a research, how to write a research question, how to write a paper. For that only, I want every state branch to have research methodology not once, at least twice a year, so that everybody can be trained. You can conduct a workshop also. And once you have any doubt, on the central API PRF, we have an advisory board, we have a scrutinizing committee, we are mentors and guides. We can tell you how to go about. One more thing the state chapter should do, apart from conducting this research methodology, conducting the program, you should also try an approach to get CSR funds. Because every state has got industries. So you tap the CSR and you can get the CSR fund. I also want every state chapter to form the state wing, plus must have the chairman, the secretary, Also, every member who joins the PRF wing pay minimum of 200 rupees for a capital fund. Suppose in the West Bengal has got 1,000 members of API. Everyone, please contribute to 200 to 1,000 rupees as your contribution as a nucleus fund to start with. Now, coming back to our research methodology, I see a lot of people are interested in doing this. I personally thank Sambhu for having initiated this project, and I think all your state leaders and the national leaders should support the PR of research wing to conduct such methodology. And we are also planning to observe November 7th to November 14th as the research week for our APA PRF. So Sambhu, you can also do some program between November 7th and November 14th, some activity. You can conduct research methodology, or you can conduct some medical college activities, or some screening program to find out, to registry. See, many people do not have any registry in the country. If you develop one project and let us know, we can have continuous registry throughout the country so that we'll have some data. For example, you want to develop asthma registry, hypertension registry, heart failure registry. Those things also can be done. For all that, basically, you must know how to go about it. That is why the research methodology is important. I am very, very happy. I compliment the West Bengal Research Wing for having started this initiative. So you will be the role model for other states to follow. So I request you to record this and then send it to me so that I'll circulate to every state so that they will also start similar programs. And if you have any printouts, handouts, you can also give it to every member. And if anybody is interested, we released a book called Research Made Easy. In the Epicon 2025 in Kolkata, I wrote a book on research methodology. It's only a very simple book, 180 pages. Suppose somebody is interested, I can send a copy to you so that also will help you. What are the steps you have to take? With this, I once again very happy to inaugurate and bless the meeting all success. God bless you. Thank you, Sambhu. Excellent. Once again, my sincere appreciation for all of you.
Thank you, Professor Muruganathan sir, and for your blessings and guidance. We are really privileged to have you today with us in this research methodology workshop. So hopefully we will start our technical session soon. Now over to Dr. Partho Sarathy Dutt.
Thank you, sir. So moving forward, we now begin our first technical session of the day. The topic is understanding the research process in medicine, and it will be delivered by Dr. Shambhu Somrat Samadar, Secretary of PRF West Bengal, and a dynamic academician and researcher. Dr. Samadar, sir, the screen is yours.
So it is visible. My screen is visible.
Yes, sir. Yes, sir. It's fully visible.
Okay. So once again, good evening, everyone. So we'll try to understand the research process in medicine for next maybe 1 hour. If you have any question or comments, you can make that in your chat box, and Clarnet team will help us to get to your questions. So Team Clarnet, can you please mute others who are actually not speaking? Clarnet team? Clarnet team, can you please meet others? Partho, can you please do the needful?
No, sir, actually, I have already requested to Clarnet team to be co-host me, but they are not respond for my chat. So I am not the co-host, so I can't.
Please call them once.
Okay, sure, sir.
Only for this inconvenience.
ஸோ நான் என்ன நீ பண்ண ரகல அவங்க கூட நான் தகராறு உங்க கூட தகராறு 75னா வாயை மட்டும் அப்படியே போன்னு சொல்லியிருப்பான். நீ 25 இருந்தா அவன் உன்னை திருப்பி கொடுக்க சொல்றான். நானும் இதுக்கு மேல கொடுக்க மாட்டேன்னு சொல்றேன்.
Who is there from
No,
Clarnet?
no, no.
I think
அது
there is
நான்
someone
கவர்ல போட்டு
from
கொடுத்தா கூட
Ringo,
நீ
I
வந்து
think. Can you please
ஓப்பன்
do the
ஓப்பன்
needful
பேப்பர்ல கொடுத்துருக்கேன்.
to mute
சார் இல்ல சார் என்னன்னு
except
தெரியாது
those who are
2 பண்டில்
speaking
இருக்குது.
and make the partition to the other as
25
per
வாங்கியிருக்கேன். அப்புறம் அவனை கூப்பிட்டு இந்த மாதிரி தான் தப்பு சார். நான் 25 கொடுத்தேன். மேனேஜர் அதனால 1,00,000க்கு 25 கொடுத்தா கோவம் தான் வரும் அவனுக்கும். அதான் கொடுத்துட்டு வான்னு சொல்லிட்டான். எனக்கு கோவம் கூட 75 கொடுத்து நீ மறுபடியும் வேணும்னு கேட்டா சரியா காது விழுது சார். விழுதுல என்ன கையில பாத்துக்கல பணத்தை. நீங்க சொல்லும் போது 75 சொன்னது 75 கேட்குது. கொடுத்த பணத்துக்கு அப்புறம் நான் வந்து திருப்பி உள்ள ஆட் பண்ணா அவங்களும் தயாரா இருக்கும். இந்த 25 சொன்னா கொடுத்துட்டு வாழ்ந்துட்டேன் அவன் அப்படிதான் சொல்லுவான்.
பேசுனீங்களா மறுபடியும்?
5 மணிக்கு மேல கொடுக்கணும்.
சொல்லுவானா என்ன தெரியல.
ரஞ்சிதா விட்டு பேசிட்டோம். இப்படி பண்ணிட்டான். இதெல்லாம் தம்பு கொஞ்சம் கமிஷன் உண்டு. அதனால நான் டைரக்டா பேசுனதுப்பா இதே அதிகம் அப்படின்னு அவன் 25 நிமிஷம் அப்படி பேசிருக்கான். அவனுக்கு பணம் கொடுக்கணும் பணத்தை வாங்கி.
So, am I audible?
Yes, Ripa, please unmute others except the speaker.
Okay, sir.
70 தான் கொடுக்கலான்னு.
So there is only 5.
வந்து 75 கொடுத்தான்.
Okay.
So the thing is that Clarnet, hello, can you hear us, Clarnet?
Yes, sir. Yes, sir.
So please be a little bit cautious. I don't know whether you are not, why you are not online always. When you have to be moderate, you have to be a little bit serious, right?
Yes, sir.
We were asking you for a long time. We are not getting you. This is not right, na?
Yes, sir.
So all the respected delegates who are present today, I am really sorry for this technical glitch. And let's start today's first presentation, that how to understand the research process in medicine. And I think all of us agree with this, why research matters. First, when we talk about research, it drives evidence-based practice. And when we talk about evidence-based practice, let's again revisit the definition of evidence-based medicine. And in the evidence-based medicine, there is 3 arms in that triangle. One arm is the experience of the treating physician or the expertise of the physician that matters. One arm is the patient response, patient experience. But the third most important part is the available evidences, scientific evidences we have. So evidence-based practice had been driven by research, and of course it improves patient care. Research guides treatment protocols and help us to develop clinical guidelines. It informs risk stratification and also where we should allocate our resources. So this is very important from the public health perspective also. And of course, research empowers clinicians to ask, to answer, and to interpret clinical questions. Hope my slides are changing. Right? So
Yes, sir.
when we talk about overview of
medical research process, we can divide that into two main stages. One, planning stage, and number two is the action stage. So when we talk about action, it requires a systematic approach, a diligent design, and robust methodology. The valid results, when we talk about valid results, it depends on one, that is minimizing bias, and two, that is maximizing validity. Again, if we talk about this validity maximizing, it could be internal validity or external validity. When we try to increase the external validity, there is a possibility that internal validity could be compromised. At the same time, when we try to increase the internal validity, the external validity could be compromised. So we need to make one balance between these two, and depending on the research question, we need to maximize the validity of the results. So there are different ways of knowing, and there could be multiple scientific methods. Why we are talking about scientific method here? Because we need to distinguish between science and pseudoscience. And these scientific methods are nothing but certain systematic observation, systematic experimentation, hypothesis formation, and testing. And therefore, when we talk about research method selection, it depends on hypothesis, study objectives, who are the population, study population, and the controls, if we are taking controls, and of course, the variables. And grossly, if we want to divide medical research, it could be primary research or it could be secondary research. The primary research, it could be basic laboratory in vivo, in vitro studies, or it could be clinical research. And when we talk about clinical research, it could be observational studies, it could be clinical trials, and the epidemiological research also comes under the clinical research. Large epidemiological studies always help us in public health decision making. Now coming to secondary research, we can have a look into the primary research data, and then we can synthesize something new from analyzing that data from the primary research. And very commonly we do the systematic review and meta-analysis, which is nothing but a secondary research. So if we talk about stages of clinical research, broadly, it could be this two stage. One is planning stage and another is the action stage. And if we talk about the planning stage, which starts from this identifying the problem. So identifying the problem means we need to know in our setup where the problem lies, what is the niche area. So mostly it could be some observation also in your clinic, that you are seeing some specific type of changes with one specific drug, and then you need to know about it more, and for that you need to do a review of literature. And after reviewing multiple literature that is available, you need to develop a research question and hypothesis. So then, after developing the research question and hypothesis, you need to choose the type of the study, the design of the study, and you have to define the target population too. The ethics is very important. Before conducting any clinical research, you should take ethics committee approval, you should prepare the informed consent, and you should follow the rules of informed consent process. Then you have to plan for data collection and statistical analysis. Now, when we talk about action stage, already we prepared the methodology. Now we need to implement the methodology. And sometimes we need to do the randomization, blinding. These are very important tools. So when we are conducting clinical trial. And after that, we need to do the data collection, then analysis, and finally reporting. Before going to the classification of clinical research, I think many of us had gone through 11 key steps of research process. Just revisiting those 11 steps. Number one, formulating the research problem. So we need to identify and define the research question or problem that needs to be addressed. For an example, if I am getting patients whose urine routine examination is showing some RBCs, and that patient was on an SGLT2 inhibitor called dapagliflozin. So then we need to think about something and we need to search whether the dapagliflozin is causing urine RBC positiveness. Then we need to do the review of literature, the second stage of the research process. What is that review of literature? We need to conduct thorough review of existing literature and research related to the topic to understand what has already been studied and discovered. So whether there are more reports which suggest dapagliflozin can cause urine RBC positiveness. We can get RBCs in urine RE when the patients are on dapagliflozin. Whether the other type of bladder cancer has some correlation with dapagliflozin molecule, what are the evidences we have? So we need to be thorough in conducting the review of existing literature. That is the second part. Now, developing the hypothesis, the third important step of that 11 steps. What is that? Creating a clear and testable statement that predicts the relationship between variables in research. So using dapagliflozin can cause bladder cancer. Whether this could be a hypothesis or either else. So I think we have a dedicated session on hypothesis development and testing. I'm not going in that part, but the fourth part is again important, the research design. Planning the overall structure and approach of the study, including selecting the research methods and data collection technique. So if I just plan that, okay, I am getting dapagliflozin patients who are having urine RBC positive. So I will take this type of patients, and then I can publish one paper that out of my 100 patients, 5 patients were on dapagliflozin and they had shown urine RBC positive. So will it be a well-researched design? I think no. So according to the hypothesis, we need to plan whether we will take certain comparator arm, that we need to decide, whether we have the facility of doing this research prospectively or retrospectively, or only via the record we have, or whether we can do it in a cross-sectional way. This all depends on my research question and the hypothesis. Then the fifth step, sample design. So determining the sample size and selecting the participants or subjects that will be the part of the study. So inclusion, exclusion criteria. These are the important steps of research process. Then after these all, we need to do the data collection. So gathering different types of data, of course, that should be previously decided in the methodology section. So whether it could be a survey-based data collection, interview-based data collection, or we are doing certain experiment and after that experiment we are collecting data. So this all need to be done. The sixth step is, we can say, the data collection thing, the data management part. Then the execution of the project, the implementation of the research plan and collecting the data as per the designed approach. That is the seventh step. We are executing our project. Then after collecting the data, the eighth step is the data analysis. So analyzing the collected data using appropriate statistical or qualitative technique to draw meaningful conclusions. This is the eighth step. Then the ninth step, that is the hypothesis testing. So we had developed hypothesis. Now we need to evaluate the hypothesis based on the analysis to determine whether it is supported or we are rejecting the hypothesis. So this is the ninth stage. Now coming to the 10th step of research process. That is the generalization or interpretation. The result of my study, whether I can make certain broader connections and interpretations of the findings in the context of the research problem. Now the question arises on internal validity or external validity. So this is the 10th step where we are talking about the interpretation of the research results, research outcome, and its generalizability. And finally, the recommendations, conclusions, presentations of my research finding to different conferences, publication, all this come in the 11th step. So I think this is very important. These are very important steps of research process, and we must maintain objectivity, rigor, and ethical consideration to ensure the validity and reliability of the results. So each step we had discussed contributes to a comprehensive understanding of the research topic and the generation of new knowledge in the field. Coming to the classification of the clinical research. So it could be quantitative clinical research or it could be qualitative. So when we talk about quantitative clinical research. Generally, we do quantitative clinical research in our thesis, in our day-to-day practice. Qualitative research, not commonly done, but it is also very important. We have our chairman of Physician Research Foundation, Professor Uttam Pal, is very famous in the field of qualitative research. Qualitative research is something which depends on focused group discussion. So here we may want to know in a certain village why those villagers are not going to the local primary healthcare center. Then we need to make certain groups, maybe each group can contain 10 to 12 persons, and it could be 2 or 3 groups. We may have a interview with them, and that interview, the results, the qualitative analysis can help us to formulate the answer, what improvement need to be done in that particular PHC, so that we can get the attendance of the villagers when they are sick. Now, research could be descriptive or it could be analytical. So, for descriptive, we could have certain case reports, we could have certain case series, or it could be certain surveillance studies. But when we talk about analytical research, yes, there could be observational analytical study. Classical examples are case control or cohort or cross-sectional studies where we are doing analysis. Or it could be experimental studies. Experimental clinical research, either it could be randomized controlled trials or quasi-experimental studies. Let's go back to the first documented clinical trial, and on this memory only, we are observing the International Clinical Trial Day, ICTD, in every 20th May every year. So this is the treatise of scurvy. And I think we know this story of HMS Salisbury, where 12 sick sailors, they were divided into 6 pairs. Those pair which had received 2 oranges and lemon, that actually worked. And ultimately, this experiment had been done. You can see the date was 1747, but it took almost 50 years. And then British made citrus compulsory in the diet of sailors, because scurvy was that time the more killer to the British Navy people than the French navies. So they need to stop this atrocity associated with scurvy. Then also you can see they took almost 50 years to implement that result outcome. So if we talk about this Lane's treatise of scurvy, very important finding we can see. Number 1, all subjects, they were in similar conditions. So like was compared with like. And now we are talking about the variable controls, the baseline control. We check the p-value at the baseline. So next important part, with every research, there is a possibility of association with confusion. So confusion could be a confounders in any research. And what happened? For the expense and perishability of the fruit, they had utilized one complicated method to prepare an inspissated juice. And now you can assume, if you are making inspissated juice, so they are actually devoid of vitamin C. And that is why that was not being useful to treat scurvy or to prevent scurvy. And that is why they become confused, and it took almost from 1747 to 1790s, then they make compulsory of using citrus fruit. So the confusion is important, and you need to solve that confusion with research only. One thing is very important. What they had not done, the review of literature. And I think this is why in our approach to research, the review of literature is extremely important. We can see the Vasco da Gama in his literature, he had instructed to bring oranges. Richard Hawkins, again, he had implemented that in the Brazil, the sailors should use oranges and lemons. So what is important, the take-home message from here is, we should have gone through the previous documentations while conducting clinical research or clinical trials. Another important issue, in a byproduct of this particular Linz study, they had distilled the fresh water from seawater. And that is one of the very important technological advances they had developed, and which also emphasize the importance of occupational health. So this was the secondary outcome of that particular research we may say. So this is again important to understand. From one clinical research, maybe we got some primary objective met, but apart from primary objective, if we are getting certain secondary outcome, that will help us to reach a new research question. Again, if we want to revisit a very famous trial, landmark trial in the history of medicine very recently, the EMPA-REG trial, what is important? Yes, these results, maybe many of us would be excited to see this dissociation of these two curves, the red line and the gray line, the benefit of empagliflozin. But before reaching to this part, what is important, and that is again the important message from the Linz treatise, that we should compare or we should control the variables. And that is why in each research, the baseline characteristic we need to check. And we need to check whether there is no significant difference between the placebo group or the empagliflozin group. Otherwise, if baseline is something different, so the whole results could be difficult to interpret. So baseline characteristic matching is very important. This was again after the EMPA-REG trial had been published in NEGM. This was the another publication of the same group, where they had emphasized the secondary outcome. And this is not from the original EMPA-REG trial, that report. This is another publication, the empagliflozin and the progression of kidney disease in type 2 diabetes. But just think about it. They had clearly mentioned it is a secondary outcome of the EMPA-REG outcome trial. Now, as we had mentioned, the importance of the secondary outcome from the Linz treatise also, they had clearly mentioned this data from the post hoc analysis. Why this post hoc analysis is important? Because many of us who cannot do the primary research, who can think about this post hoc analysis, and here it is a post hoc renal composite outcome by which they were showing the empagliflozin could be beneficial in improving renal outcome. Just I want to stop here for 1 minute or 30 seconds. Please think of yourself and choose the right option here. What is the importance of post hoc analysis in any scientific research? And what caution should be exercised in its interpretation? The fourth option, post hoc analysis, is primarily used for confirming hypothesis and established by experiments, and its results are always definitive. Number two, post hoc analysis is crucial for generating hypothesis from existing data, but its conclusion should be viewed with skepticism until confirmed by further prospective studies. C, post hoc analysis is irrelevant in scientific research as it does not involve experimental methods. I think many of us will not agree with this. D option, post hoc analysis is the only method used for determining some structural aspects in molecular biology. I think I hope you are not agreeing with, maximum of you are not agreeing with this D option also. And yes, I think many of us would agree that post hoc analysis is nothing but an important tool to generate hypothesis. It is a hypothesis generating analysis from existing data, but the conclusions should be viewed with some certain skepticism until we can confirm. So this will give us a new research question. And as we were talking about that secondary post hoc analysis of from the empiric data, ultimately again another publication in NEJM, the EMPA-Kidney, the famous another landmark trial. So from empiric to another EMPA-Kidney. So two landmark trial, one bridge was the post hoc analysis. And this is the beauty of clinical research. And we can see this particular prospective study, we can say that experimental study, the randomized control trial, had confirmed the finding which were there in the post hoc analysis. Coming to an important zone, because we need to do study and we need to understand what type of study we are going to conduct. And this is the very basic question we need to ask at the beginning. As investigator, whether I am assigning the exposures, I am deciding which arm will take the study drug and which arm will take the placebo or the other drugs. If I am assigning the exposure as an investigator, then it should be experimental study. If it is not, if I as investigator not assigning the exposure, then it is an observational study. So when it is experimental study, whether the allocation is random, it is randomized control trial. If it is not, then non-randomized control trial. For observational study, if there is certain comparison group, if it is the answer is yes, then we are doing some analysis. So it is an analytical study. If it is not, we are only giving some description, just like a case series or reporting a group of my patient from my clinic, then there is no analysis, analytical information. We are just describing the demography, describing the clinical finding. So it is a descriptive study. When it is an analytical study, we need to ask ourselves whether or in which way the direction lies. If we are moving from the exposure to outcome, then it is a cohort study. If we are going back from outcome to exposure, then that is a case control study. And when we are doing a snapshot-like thing where the exposure and outcome at the same time we are capturing, then that is the cross-sectional study. So another way we can say, though that is this thing we need to be very cautious while using, retrospective study, prospective study, and there could be a possibility in some subgroup of the studies which could be ambispective. That means in a certain position, we are going back to take certain previous data of the patient, and then we are coming to today's point, and then we are prospectively observe our patient. So both prospective and retrospective is there. So that could be considered as ambispective design. And I think this chart is important for all of us to understand, because all of us are doing something with drugs, and as we are conducting something with drugs, we need to know the journey of the drugs from the conception to the patient utilization. These phases are important, which starts with certain discovery phase or preclinical studies, then the clinical studies, phase 1, phase 2, phase 3 clinical trials, and then when they got approval, we will have phase 4 trials. And when we talk about phase 4 trials, they are regulatory trials. It is not that all the post-marketing surveillance studies, they are phase 4 trials. They are not phase 4 trials. If I am doing some observational studies, this need to be understood. And if we are conducting any of these phase 1 to phase 3 trials or phase 4 trials, we are bound to our regulators, because these are regulatory trials. So in nutshell, when we talk about phase 1 study, phase 1 trial, they are generally for the safety and tolerability, to know the pharmacokinetics for a particular drug. Phase 2 trials, generally we want to find the dose. This is a proof of concept study. Both the pharmacokinetic, pharmacodynamics come here. And then finally the phase 3 trials, where we want to check the clinical efficacy and safety outcomes. When we talk about phase 4 study, phase 4 trial, they are more pragmatic. There we want to see the effectiveness, not the efficacy. The difference between efficacy and effectiveness, the efficacy, there is more internal validity. So they are the clinical trials of this type of phase 3 clinical trials. They will have much more internal validity. But when we talk about phase 4 trials, they are effectiveness trial. They will have more real world generalizability. So more external validity in effectiveness trials or phase 4 study trials. These are few numbers. If one drug I am taking and that enters into the phase 1 trial, so there could be a 10% success rate to achieve the approval of that particular trial. For one drug to pass from the phase 1, that is 65% chance. So most difficult period we can see the phase 2. So 30% success rate in the phase 2. And again, once it enters into the phase 3 clinical trial, so there is a chance of 60% success. One important area we need to know while doing clinical research, especially when we are conducting trials, to minimize the assessment bias in different trials. And blinding is a very important way to minimize this bias, especially the assessment bias in trials. When you talk about single blinding, it involves masking the subject only, double blinding, masking the subject and investigator, triple blinding, it extends to data analyst also. So along with subject and investigator, data analyst will be blinded. And when we talk about unblinding, this unblinding challenges arise in different trials with certain specific limitations, especially when we face certain safety concerns. Now, the final part of my presentation, because when we conduct clinical research, we need to know these histories, these atrocities. We can see here, I think my cursor is visible here, the Nazi war crimes. The research participants were exposed to severe cold, severe hot, and just the experimenters, very sad, we cannot tell them doctor today. They were just wanting to witness what are the physiological changes that happened with extremes of temperatures until these research participants died. This is the picture of Tuskegee syphilis trial, where the black American prisoners, they were being exposed to syphilis and they were not being treated syphilis for syphilis. There was availability of penicillin that time in US, but they were not being given the penicillin option. Just they were being observed what will happen to them with syphilis, the natural history of syphilis. These doctors want to see. And here we can see this school, you may think this is a beautiful school. But what happened there? They had taken participants from their students who are mentally retarded and they were being exposed with hepatitis A just to see the natural history of hepatitis A. Here the question on vulnerability arises, vulnerability of research participants. And I think this sweet baby, unfortunately, there is no limb, and this is called as hocomelia. This had been first time observed by this great doctor, Austrian doctor, McBride. He had noticed that some of his patients, the mothers who were on thalidomide, they were giving birth to this type of babies. So he had noticed, and then he had reported that. And after his report, the thalidomide had been banned, especially using in the mothers. And this lady, Kelsey. So Kelsey, you are being, we can see the honored with the highest civilian award from the president, Kennedy, that time, because she had prevented the entry of thalidomide in US. She had noticed something which is not appropriate. Certain animal studies she had noticed and prevented the entry of thalidomide in US. So this massacre was not there in US. So she was being awarded with the highest civilian award. And after that, we got something called as Kefauver-Harris Amendment. Again, this was a fake test subject. So subjects were not being told they are subject, but certain cruelties had been experimented. This was psychological experiments they had done. So why these incidents, atrocities are important? Because with this type of hammering in the patellar knee, patellar tendon of knee, there were certain knee-jerk responses, and we got this ethical principle which had developed in the field of clinical research, be it the Nuremberg Code after the Nazi war crime, then the Declaration of Helsinki, then the Belmont Report, and we had discussed about Kefauver-Harris Amendment in 1962, which had strengthened the federal oversight of drug testing and included a requirement for informed consent. The Nuremberg Code was very important because they had first time mentioned about the essentiality of voluntariness of consent. Then when we talk about Helsinki Declaration, they had given us certain general principles and specific guidelines on use of human subjects in medical research. And the famous Belmont Report, it told us about three important issues. Number one, we need to respect for persons, that protects the autonomy of the individuals, special protection for those with diminished autonomy, vulnerable population, beneficiaries. We need to maximize the possible benefit, minimize the possible harm. So beneficiary and non-maleficents. And then the justice, the equitable distribution of chance of being participated in a specific research. So subjects should be selected fairly to ensure that the benefits and burdens of the medical research are evenly distributed. We have our regulatory body like CDSCO here. We have our ICMR who are developing guidelines to facilitate clinical research in India. And as we were talking about this design, we need to understand, if I want to do an experimental study, I need to be thorough with the document New Drug Clinical Trial Rule 2019. And for observational study, this is must. The National Ethical Guidelines for Biomedical and Health Research involving human participants, the ICMR 2017 documents. These are two important issues, especially when we talk about New Drug Clinical Trial Rule 2019, which had introduced ethics committee registration, compensation norms, post-trial access, and we are talking about ICMR guideline, which had given enough emphasis on informed consent, vulnerable population, and risk benefit. So these are the core ethical principles we need to understand before doing any clinical research: autonomy, beneficients, non-maleficents, justice, that is fair participant selection, and of course, now we are talking about privacy, the data security and confidentiality. Most importantly, we need to know this: responsibilities of key stakeholders in clinical trial. When we are talking, we may involve multiple other stakeholders, but we often forget the research participants are also an integral part of these stakeholders. So we do research for the pupil, and that should be with the pupil, not on the pupil. And these are a few important stakeholders if you want to do clinical trials. The relationship between sponsor, CRO, investigator, site administrator, IRB, IEC, or regulatory authority. That should be transparent, and there is a clear-cut guideline directed relationship that is available. And if we need to go through the New Drug Clinical Trial Rule 2019 and ICMR 2017 guideline. I want to end here with this very famous quote by Vidura to Yudhishthir, because this is the essence of any clinical research ethics. When we talk about ethics in anything, it is a multiplied product of two M. One M is for morality, and another M is for materialism. So it is a multiplied product of these two M. Now, materialism is important bad science is equal to bad ethics. There is no other way in that. And something multiplied by 0 is always 0. So ethics will be 0 if it is a bad science. But again, we need to think the morality one, because while doing clinical research, we need to ask ourselves this particular question. Atmana pratikulani pareshuna samacharet. That means do unto others what you would like others to do unto you. So as a researcher, as a clinical researcher, we need to place ourselves in the chair of the research participants. And then we need to think whether my research is okay or not. That question, if we ask and we get some answers to that particular question, if that answer is yes, the research is moral. We can go ahead with morality. And of course, when conducting clinical research, we need to remember we may see some negative results in my clinical research, but that is not. We need not to stop on that. We also need to publish our negative results, because that is also important for the society, for the bigger community. And that is why we should learn from Krishna, as he had told, Yogastha Kuru Karmani Sangam Tattva Dhananjaya Siddho Asiddho Samo Bhutva Samattang Yoga Uchate. So this principle of equanimity, that is very much integral part of clinical research. We could have certain success or failure, but we need to be transparent and we need to accept that. So thank you. I think that was the last slide of mine. And I think this was something which I want to share with you all on the basic of clinical research. And over to Dr. P.S. Datta. If we have some question or comments, we can take that later, because Sujoy sir has already joined.
Yes, sir. Thank you, Dr. Samadhita sir, for that clear and comprehensive overview of the research process. And this is a perfect start to our academic journey. So thanks once again. And I guess Dr. Sujoy sir is here with us, and he is our next speaker. So our next session is into one of the most crucial steps in the research journey, developing a research question and hypothesis. And we are privileged to have Dr. Sujoy Ghosh sir, Professor of Endocrinology at IPGMER, Kolkata, to enlighten us. Yes, Dr. Ghosh, we look forward to your session. Over to you, sir.
Thank you very much. Hopefully I'm audible and the
Yes,
slide
sir.
is visible.
Yes, you are audible and your slide is visible too, sir.
Big thank you to PRF and API for having given me this opportunity to interact with all of you. After a very detailed presentation by Shambho, I think my session is not going to last for 1 hour. I'm not going to bore you that long. But like Partho said, that this bit of the session is actually very important. There are two things which I, as a clinician, have seen when in research people go absolutely wrong. Number one, people usually do not do a very good literature review before they start the research. So there are questions that are already answered, which people try to reinvent and redig. That's number one. And number two, the students particularly are confused. Why am I doing this research? Half of the time I see most of the dissertations that we see is more like a fishing expedition, in which people have tried to collate X amount of data, and then depending on the results of the data, they're trying to figure out at the end of the study what's positive out of that study, as if that was the research question to start with. So one of the great researchers made a very, very valid statement. He said, we don't need so much of research. Every year, so many papers are getting published. We need less research, but we need better research and research done for the right reasons. It should not be done just for the sake of a dissertation for the exams. It should not be done for the sake of a publication so that the faculty can get the next promotion. Unfortunately, in India, these two are the main drivers of research, at least in academic institutes. Now, if you're looking at research, you have to understand that there are basically two different kinds of research. One where you're trying to look at certain aspects relevant to advancing of knowledge. So, for example, GLP-1 and GIP, they were discovered almost about 50 years ago, 60 years ago. And in fact, the first incretin studies was done somewhere in the 1920s. Now, that knowledge at that point of time was advancement of knowledge. It did not lead to immediate application. On the other hand, for example, today Axiom 4 has been launched. And I don't know how many of you know, there is an Indian gentleman on that Axiom 4. And of all the researches that is going to happen there, one is to look at insulin, the bioavailability of insulin, the stability of insulin in space, and ISRO is doing a research project in that Axiom 4 in terms of looking at bone health and weightlessness and how you can actually reduce sarcopenia. Now, all of us are using GIP and GLPs, and we are knowing sarcopenia is a reality. Now, if this research comes up and tells us that, if you do X, Y, and Z, you reduce sarcopenia, there will be immediate application of that because that's already a burning problem. So therefore, excuse me. Therefore, we have to try and see, either we try to go for low-hanging fruit, wherein you don't have to put in a huge amount of effort, but the research will be immediately impactful and will be advancing knowledge. That will be what we call in the dull quadrant. Whereas we should try and avoid wasteful quadrant. That means something which will only add to my knowledge, but which will not help in application or translation. So therefore, whenever you are thinking of doing a research and thinking of a research question of whatever sorts, try to think of the translational value of things. The major challenge in India is we are doing too little translational research. So if you have heard the director of ICMR, DHR in recent times, they have said enough is enough. We are not going to waste money doing certain obscure research which will lead to few publications in journals, but which will not help improve the health or improve outcomes in our patients in India. Because it's, after all, taxpayers' money. So priority definitely is there in translational research. So all of you are aware of the Max Planck University in Germany. And he said, an experiment is a question which science poses to nature, and a measurement is the recording of the nature's answer. So scientific principles and laws do not lie on the surface of nature. They are hidden and must be wrested from nature by an active, elaborate technique of inquiry. So if you're looking at a scientist or a researcher, who is he? The scientist is not a person who gives the right answers. He's the right person who asks the right questions. So this brings us back to our topic for today, that a true scientist is one who is able to ask the right questions in research. And asking the right question only will reveal the right answer. And in most of the cases, the answer is there. The revelation of the answer depends on the question that actually you ask. So when you're talking about a research question, the details of the problem statement have to be there. You have to further describe and refine the issue under the study and focus to the problem statement, guide data collection and analysis, and set context. For example, a research question is a statement that identifies the phenomenon to be studied. Does material X123 affect fatigue strength to prevent fracture under cyclic loading and ductility? Something like that. Very obscure. So your research question has to be very clear. It has to be very focused, concise, complex in terms of the aspects that it deals with, and arguable. So I'll give you a few examples. We all say, how should social networking sites address the harm they cause? That's a very vague, unclear question. A clearer question would be, for example, what action should social networking site like MySpace and Facebook take to protect users' personal information and privacy? An unfocused question would be, what is the effect of environment from global warming? A more focused question would be, what is the most significant effect of glacial melting on the lives of penguins in Antarctica? So you have to decide whatever I'm asking. It can't be too open-ended. It can't be too vague. It can't be too big. And remember, the best of studies actually answer one question, and the summary of the best of studies is only one line. So if you can change one line in the textbook or in the management guideline or protocol, that's a good research that you've actually done, because the research question was very pointed and the answer therefore is very clear-cut and focused. So in terms of the research question, does the research question provide a clear idea of what the research project is about or what it hopes to address? Can it be addressed by research? So the possibility and the plausibility and feasibility of that question is also important. Is it possible to deduce from the research question possible answers or modes of addressing the question? Feasible fieldwork in terms of sampling and data collection. Is the question interesting, relevant, and important? Does it emerge from a broader set of disciplinary or professional issues? These aspects are important. So let's try and figure out one such example. For example, I might say, okay, if you're driving particularly on the countryside, you will see chickens crossing the road. So why do they cross? So devise a purpose statement. The purpose of this study is to determine the cause of why chickens cross roads. So one version of the research question could be, why did the chicken cross the road? This is too broad, not specific enough, relies on qualitative data which may not be attainable. You can't ask a question to the chicken because the chicken doesn't interview well. So the second option could be, how many chickens crossed, say, a particular road, MG Road, in the period of January to July? This is too specific, closed information, answer in a stat or a number. The third version could be, what are some of the environmental factors that occurred in Kolkata between January and July that would cause chickens to cross MG Road? So you are talking about now specifics, measurable, investigatable, and gets into the why of the issue. Let's try something else. A broad topic. Can we use hydrogen fuels in your aeroplanes? And how? So the devise a purpose statement. The purpose of the study is to determine the use of hydrogen fuels in aeroplanes to find a greener way to fly. So one research question could be, what are the best alternatives to existing fossil fuels in aeroplanes? Pretty broad, right? The second version could be, does hydrogen fuel in aeroplanes reduce the sulfur dioxide emission? The other version could be, does replacement of fossil fuels by hydrogen fuels in aeroplanes result in zero emission flying? So you are modifying, sharpening your questions to actually decide upon what you really want from the study. So I'll give an example. For example, in cardiology, a study on ischemic heart disease. So you want to find, the goal of this study would be to contribute to prevention of ischemic heart disease. So the primary objective may be to determine the effect of reducing LDL cholesterol on the occurrence of MI. So the research question here would be, does agent A decrease the risk of MI? The research hypothesis would be the risk of MI among patients treated with agent A is lower than risk among controls not treated with agent A. So here again, you can see, first I've told you what the research question is. And that research question essentially is based on a research hypothesis. So even though the two are interlinked, they are a little bit different. So in the hypothesis, you first make an assumption, or you think that this is what you think is right. And the research question then tests that research hypothesis. I'll give you another example. The human papilloma virus subtype 16 infection, does it influence the development of oral cancers? There you have to think of all of those bits. So the format that we often use is what is called the PICOT or PICOT format. The first P would be participants. So you have to tell us who that particular group of people are. Who are your participants? Is this boys? Is this girls? What age group? Specifics about inclusion, exclusion that you want to treat. The intervention. Say, for example, in a statin trial, they'll be either exposed or with having the control. The comparators, there is always an alternative, another therapy, and outcome and the time frame over which you expect a result. So let's look at an introductory case. A 45-year-old male with hypertension presents to your clinic for follow-up. At a prior visit, he was screened for diabetes. His A1C was 7 on 2 occasions, which is a new diagnosis of diabetes. Blood pressure is 135/80. He has no evidence of microalbuminuria or kidney disease. You may recall ACE inhibitors are good for preventing diabetic kidney disease, but you're not sure if this fact applies to this particular patient. So how can I develop a clinical question? So think of the PICO question. So PICO, the first P is the population. Starting with your patient, ask how would I describe a group of patients similar to mine? Be precise and brief. So here you would probably think a type 2 diabetes individual with hypertension, maybe male, maybe some duration, and maybe even control of or age group and something like that. So you have to define which patient group. Then you're talking about intervention or comparison. Ask, what is the main intervention I'm considering? Is it an ACE inhibitor? And what is the main comparison or control group? People who are not getting an ACE inhibitor or people who are on other antihypertensive drugs. Be specific, but consider feasible alternatives. In today's world, for example, if you said, oh, I'm not going to give any other drug to the patient, I'm just going to give the treatment group an ACE inhibitor and the control group I'm not even going to treat. Ethically, you cannot do that. And then think of the outcomes. Can I hope to accomplish, or what could this exposure really affect? Select patient-oriented outcomes instead of numbers. I'll come to that later. So for example, if in such a trial you're thinking of outcomes in terms of death, in terms of end-stage kidney disease, then you imagine how many people you will actually have to enroll and for how long you will actually have to do that trial to get that outcome. On the other hand, if you're satisfied with, say, a change in albumin creatinine ratio, that might happen within weeks of the treatment that or intervention that you're talking about. So outcomes you have to think of very specifically. So the P, the problem of the population in adults with diabetes mellitus and hypertension. Intervention is, does an ACE inhibitor. Comparison, either you are thinking of compared to placebo, or in most cases what that we will think of is the beta blocker, the calcium channel blockers, and the diuretics. And the outcome could be prevent development of microalbuminuria or prevent progression of EGFR. So you've understood the same question, you've refined it in terms of this PICO approach. Now, imagine I'm just talking about these outcomes that we discussed. If I go back to a couple of slides here, just let me get back here. The last slide. most of the time, we as doctors, we are often thinking of numbers. We are saying, okay, how many are going to progress in terms of lowering of EGFR? How many is macroalbuminuria going to come down to microalbuminuria? Or the LDL cholesterol is going to come down from 150 to 70. Now, honestly, do you think these are numbers that we are worried about as treating physicians? We are definitely concerned about this as treating physicians. But what about the patient? Are the patients really more concerned about this? Or is he concerned about something beyond these numbers, what we call patient-oriented outcomes? These outcomes, if you're talking about, you have to now think of what matters to the patient. So we mostly think of disease-oriented evidence, measure outcomes that are markers for disease. We say, the liver fat has decreased, the fibroscan has improved. We are saying LDL cholesterol numbers have improved. We are talking about urine albumin creatinine has improved. But the patient-oriented evidence that matters might be very different. He wants to know, my father went into dialysis. Will I need dialysis as well? Will I progress to end-stage kidney disease? Will I have a heart attack? Will I die of a heart attack? What will be my lifespan? Will it be shortened? So these would be patient-oriented outcomes, which patient might think are the more important endpoints or outcomes in a study. So therefore, your clinical question you have to think of is, what are you trying to achieve in terms of the question that you're asking in this study? Are you trying to look at disease-oriented outcome evidence, or are you trying to look at patient-oriented evidence? So if you're talking about disease-oriented evidence, it could be lab values, biochemical markers, things like blood pressure. Even we are saying, we see so many papers published about what has happened to carotid intimal thickness. Do patients really care? Perhaps not. Whereas patients are more concerned about morbidity. Will I lose my job? Will my foot come off? Will I be able to still see from diabetes, or will I develop retinopathy? Will I die? And of course, quality of life is entirely something that the patient appreciates more than we do as clinicians. So I will give you examples of more of disease-oriented evidence versus patient-oriented outcome evidences, which will again determine in your research question what you are going to put as outcomes. So disease-oriented evidence, intensive treatment with lower blood glucose level in type 2 diabetes. So when the ADVANCE and the ACCORD trials were published or even planned, we all wanted to see if going down to HbA1c less than 6.5 could be achieved and whether it was safe. The patient-oriented evidence, if you look at the end of the study, intensive treatment, just the glucose control in such individual did not lead to decreased mortality. So that is what he's more interested. We are trying to say, for example, beta carotene and vitamin E are good antioxidants. The patient actually then reads up studies to say that neither beta carotene or vitamin E prevents cardiovascular disease or cancers. I'm really not bothered whether they're actually antioxidants or not. Similarly, we often actually prescribe a lot of medication even for quitting smoking. So the drug varenicline can help smokers stop smoking, which should lead to a decrease in cardiovascular events. So your endpoint could have been quitting smoking, but it has now been shown, if you're looking at the patient-oriented evidence, that it increases cardiovascular events. Again, the older antiarrhythmic drugs can decrease irregular heartbeat in a patient with asymptomatic arrhythmias, but medical treatment of asymptomatic arrhythmia increases mortality by 10%. So remember the importance of the different outcomes. So coming back again, we were talking about the patient, the intervention, the comparison, and the outcome. We had talked about worsening EGFR. So is EGFR an outcome our patient cares about? So that's a disease outcome that we are thinking of. Possible patient point of view, the patient wants to know, in patients with diabetes, are ACE inhibitors associated with lower mortality? In patients with type 2 diabetes, 2 ACE inhibitors delay progression towards end-stage kidney disease requiring dialysis. Somebody else might look at the same thing in terms of ESRD and saying, does it reduce the requirement of a renal transplant? So when you are thinking of an outcome variable, there are certain desired properties of an outcome variable in your study that you have to think of. It should be appropriate, be fitting for the objective of the study. For example, if you're looking at, say, the Carmelina study or the Carolina study, they tried to look at, say, reduction in albuminuria, tried to look at differences in grades of albuminuria. Now, if you're looking at a smaller study duration, that might be good. Others might argue and say albuminuria is only a surrogate marker. It is a function of endothelial dysfunction. If you are actually wanting to look at an outcome to say whether this is really renoprotective, you should look at a composite renal outcome, just like what Shambhu was talking about, the EMPA kidney study, the DAPA-CKD study, the CREDEN study. Next, be objective. That it should require less subjective judgment to measure. So there has to be something like that. For example, doubling of creatinine time or halving of the EGFR and things like that. And it should be valid, that it should measure that which is intended. Albuminuria per se will not necessarily tell me what the renal dysfunction is going to be in terms of progression. But a composite renal outcome will tell me what the renal progression is. Be reproducible, be precise, and be reliable. That is, should be easily be reproduced in different times, settings, and be clinically available. That it should be available as part of clinical care. If you make it very obscure, it might be very difficult. Be easily quantifiable, be efficient in terms of the time and the money that you need for the test, be sensitive, be specific, be responsive. That means it will change very easily. For example, if you're doing a study on neuropathy, and if you are going to use a monofilament test to say that whether this is good or bad, this study is going to be very, very insensitive. On the other hand, if you are using corneal confocal microscopy for the same intervention in neuropathy, it will be a much more sensitive test. And it should be straightforward. That is, should allow easy interpretation of results. So again, the classical smoker, non-smoker concept in lung cancer. This is a study which you can think of. You're looking at certain population of participants. Here, the example given is British doctors. In terms of the smoking status, measured, that's the exposure, and the outcome is lung cancer. Similarly, you can think of mammography screening, whether it's positive, it's negative. So that would be a diagnostic or a prediction study. So depending on what you are actually doing in terms of the study, the PICOT question can be modified. Coming to the next aspect, after we finish the research question, is hypothesis. So often students confuse what is the difference between research question and hypothesis. People will directly say, oh, hypothesis is what I thought of, and the research question is telling me is actually an extension of that. Partly true, but again, a recap. Hypothesis are single tentative guesses, good hunch, assumed for use in devising theory of planning experiments intended to be given a direct experimental test when possible. So you might say, okay, use of a GLP-1 might actually improve amongst a lot of metabolic complications. Maybe some obscure complication like, say, erectile dysfunction will improve if you're using a GLP-1. That's your gut feeling, it's telling you, and your hypothesis is GLP-1 improves erectile dysfunction. So a hypothesis is a conjectural statement of the relationship between two or more variables. Here it is, say, people with obesity getting the drug will improve is the hypothesis. And it's often useful to write down that statement of hypothesis that you're thinking of. It's a formal statement that presents the expected relationship between the independent and the dependent variable. A research question is essentially a hypothesis asked in the form of research. So for example, I say my hypothesis is GLP-1 inhibitors or GLP-1 agonists improve erectile dysfunction. That's my hypothesis. The research question would be, would, for example, GLP-1 injectable semaglutide used in individuals with obesity with no other comorbidity improve erectile dysfunction as expressed by FSFI questionnaire by a 2-point improvement or something like that. So you are specifying the population, you are specifying the intervention, you are specifying a comparator, and you are specifying an outcome. So that is how from hypothesis you are going to a research question. So the nature of the hypothesis, let's talk about that. Hypothesis is a clear statement of what is intended to be investigated. It should be specified very clearly before the research is conducted and openly stated in reporting the results, which allows to. Now, I have seen very poorly drafted manuscripts in most of the circumstances for students. When you're writing the introduction of any research paper, original research paper, please do not start by saying, India is a country which has got the highest number of type 2 diabetes individual, and then quote the ICMR INDIAB study, and then say, all of that is not what is actually interested, the reviewer is interested in your research introduction. What he wants is, with regards to what you are going to address in this paper, what is already known, what is the knowledge gap, how have you identified that gap, and what is your hypothesis which you want to test in this study. The last paragraph of your introduction of your original research paper should actually tell us in very clear words the hypothesis that you are testing, followed by the research question. That means in whom you are going to test it, this hypothesis in the PICO format. Then the reviewer clearly knows that this is what this guy wants to try and tell me in this particular study. A problem cannot be scientifically solved unless it is reduced to hypothesis form. It's a powerful tool of advancement of knowledge, consistent with existing knowledge and conducive to further inquiry. The nature of the hypothesis. It can be tested or falsifiable. Hypotheses are not moral or ethical questions. It is neither too specific nor too general. It's a prediction of consequences. It is considered valuable even if proven false. So Shambhu was talking about negative results also have to be reported. So the examples of hypothesis. Health education program influence the number of people who smoke. Newspaper affect people's voting pattern. Attendance at lectures influence exam marks. Diet influences intelligence. So these are kind of assumptions that you're making. This is what your beliefs are. And then you have to frame a research question to answer such things which you think or believe is the right answer. So when we talk about hypothesis, there are two things that we have been taught traditionally: the null hypothesis and the alternative hypothesis. So when you're talking about the null hypothesis represents a theory which has been put forward, either because it is believed to be true or because it is to be used as a basis for argument but has not been proved before. It can be anything. For example, in the CV outcome trials, either you are trying to do a superiority trial or a non-inferiority trial, and you say, for example, drug like semaglutide injectable or drug like injectable tirzepatide used in patients with obesity, without diabetes, with comorbidities and complications, if used over a period of time, we believe reduces cardiovascular outcome. So that's your hypothesis. So now that hypothesis has to be tested in a clinical trial of a CV outcome trial, where you will then define the research question You will define the population, you will define the intervention, you will define the comparator, and your outcome measure is the 3P maze. And it has serious outcome if incorrect decision is made. In research studies involving two groups of participants, example experimental group versus control group, the null hypothesis always predicts that there will be no difference between the groups. So you will have to reject the null hypothesis if you want to actually show that this particular intervention is of use. The alternative hypothesis is a statement of what a hypothesis test is set up to establish. It is the opposite of the null hypothesis and is only reached if the alternative hypothesis is rejected. Frequently, alternative hypothesis is actually desired conclusion of the researcher. So, for example, in a CV outcome trial, your null hypothesis is adding GLP-1 injection in patient with obesity does not improve cardiovascular outcome. That's your null hypothesis. And at the end, you show that if you treat versus you don't treat with the GLP-1, the outcomes are different. The GLP-1 improves outcomes. So therefore, you will say that the null hypothesis is rejected and the alternative hypothesis is accepted. So let us give again examples. In a clinical trial of new drug, the null hypothesis might be that the newer drug is no better on average than the current drug. You would write that H0 there is, or the null hypothesis, that there is no difference between the two drugs on average. The alternative hypothesis might be that the new drug has a different effect on average compared to that of the current drug. You should write then H1, the two drugs have different effects on average. The new drug on average is better than the current drugs. You would write H1, the new drug is better than the current drug on average. And it is always good to write down the null hypothesis and alternative hypothesis at the very beginning of the planning of the research project. We give special considerations to the null hypothesis. This is due to the fact that the null hypothesis relates to the statement being tested, whereas the alternative hypothesis relates to the statement to be accepted when the null hypothesis is rejected. The final conclusion, once the test has been carried out, is always given in the terms of the null hypothesis. We either reject null hypothesis in favor of the alternative hypothesis or do not reject null hypothesis. We never conclude reject alternative hypothesis or even accept alternative hypothesis. Why I am making such a statement? If we conclude do not reject null hypothesis, this does not necessarily mean that the null hypothesis is true. It only suggests that there is not sufficient evidence against the null hypothesis in favor of the alternative hypothesis. Rejecting the null hypothesis then suggests that the alternative hypothesis may be true. Coming to the next bit of hypothesis that we also need to know is what is called directional hypothesis. They are usually derived from theory. They may imply that the researcher is intellectually committed to a particular outcome. They specify the expected direction of relationship between variables. That is, the researcher predicts not only the existence of a relationship, but also its nature. I'll give you a classic example. The class 10 board examinations in West Bengal. Now you can ask two different things or make two different hypotheses. The first hypothesis could be that there is no difference in the results of boys versus girls in class 10 board exams in the state of West Bengal. Now, this is a bi-directional hypothesis. Why? The girls might be better, the boys might be better, right? On the other hand, if you write a hypothesis saying that girls, I think, do better than boys in class 10 board examinations in West Bengal. So here, your hypothesis is one tail. That means you are only thinking of girls being better than boys. So you only have to prove that the girls are not doing better than boys. You don't have to prove whether the boys are doing better than something else. So it's only one directional hypothesis. You may assume that when manipulating the independent variable, there will be one specific change in the dependent variable. You can predict if this change will be positive or negative, whatever you've thought of. The non-directional hypothesis is used where there is little or no theory or when findings of previous studies are contradictory. They may imply impartiality, do not stipulate the direction of the relationship. Here's where, for example, you're thinking of boys versus girls, there is no difference, or is there any difference, whatever your null hypothesis might be. The results will be positive even if A is better than B, or B is better than A, or it's a positive to one end or a positive to the other end. So it's a two-tailed test that you will actually have to do. So now coming to the next bit, that is formulating a hypothesis. It is important to narrow a question down to one that can reasonably be studied in a research project. The formulation of hypothesis basically varies with the kind of research project that you are doing. So let us think of a qualitative process. Can be divided into a deductive study and an inductive study. Deductive studies, you look at observations, then the pattern, think of a tentative hypothesis, and then you decide on the theory. Whereas if it's an inductive one, then you already have a theory that you are thinking of. With that, you are formulating a hypothesis. Then you are going for observations and you are confirming your theory. Qualitative approach, less commonly done, but the use of research questions as opposed to objectives or hypotheses is more frequent. Use of words what or how, specific whether the study discovers, seeks to understand, explore, or describes the experiences. Use of non-directional wording in the question. These questions describe rather than relate variable or compare groups. The questions are under continual review and reformulation, will evolve and change during the study. Questions are often open-ended without reference to the literature or theory and use a single focus. This is the question that you're trying to answer when you do research on a topic and write a research report. It should be specific as possible. In some cases, you may make 2 or more research questions to cover a complex topic. For example, if you're studying the effect of sleep on reflexes, you might formulate the following research questions. What are the effects of sleep on reflexes? A similar question might be, does sleep have an effect on reflexes? Or is maximal reflex efficiency achieved after 8 hours of sleep? The goal of your research is to find the answer to your research question. So again, coming back, the hypothesis versus the research question. A hypothesis is defined as an educated guess. It might be absolutely wrong though. Because for example, Donald Trump had a hypothesis that he's going to blast off Iran within 3 days. It didn't happen anyway. But that was his hypothesis. While a research question is simply the researcher wondering about that particular issue. Hypotheses are part of scientific research methods. They're employed in research in science, sociology, mathematics, and more. Research questions are part of research methods and are also used in many fields, including literature and sociology. The hypothesis versus research question, as its name suggests, research questions are always written as questions. Hypotheses are written as statements, preceded with the word I predict. I predict that this is going to happen if you treat A versus B. For example, a research question would ask, what is the effect of heat on the effectiveness of bleach? That is, a hypothesis would state, I predict heat will diminish the effectiveness of bleach. So before writing a hypothesis, the researcher must determine what others have discovered about this subject. Good literature search. On the other hand, a research question requires less preparation, but focus and structure is critical. So you already have a hypothesis. You're now focusing and restructuring this question. A researcher using a hypothesis would look up studies about bleach, information on the chemical properties of the chemical, then heated, and data about its effectiveness before writing the hypothesis. When a research question, the researcher would think about how to phrase the question to ensure its scope is not too broad, too narrow, or impossible to answer. When writing the conclusion for research conducted using a hypothesis, the researcher will write whether the hypothesis was correct or incorrect, followed by an explanation of the results of the research. The researcher using only a researcher question will write the answer to the question followed by the findings of the research. So I think I have bored all of you enough, and my time has come to more or less wind up and then take questions, but I will leave you with a few things to think about. Knowledge is about having the right answers. Intelligence is about asking the right question. So unless and until you ask the right question, you will not enhance knowledge of an individual. So it's very important that we ask the right questions. Thank you very much.
Thank you so much, Dr. Ghosh. That was a very thought-provoking and practically enriching session. So, if anyone have any queries, we have time to take that questions or queries. So, I'm going to request the Planet team if anyone have post any questions or queries.
Right. I think we could probably break to the next session. I think which is a break.
No, actually I think there is no question as you explain fantastically. So I think there is no questions. So now we'll take a break of 35 minutes and please return by 5:15 p.m. sharp as we resume with our next session on start. And our next speaker will be Professor Nandini Chatterjee from same institute. So please return by 5:50 p.m. Thank you. Thank you all.
So in the meantime, I request Clarnet team to please send us message to all the participants that we will be back by 5:15. Can you hear us, Clarnet team? Team Clarnet, can you hear us?
Hello.
Yes, sir.
Yeah, can you hear us?
Yes, sir, I can hear you.
I think you need to be a little bit more attentive. We are calling you many times. So the thing is that when we are not talking, someone is not talking, please stop their video. Okay, this is the number one thing you have to follow.
Okay, sir.
Number two, you have to send all the delegates who are attending today one message that our next part of today's session will be started from 5:15.
Okay, sir.
So make it live in that way so that everyone can see that from 5:15 we have the next session.
Okay, sir.
And make all the non-speaking faculties who are there, so their video need to be stopped and they should be muted. Except the speaker, everyone should be muted.
Okay, sir. Okay, sir. And the excuse me, sir. Hello. Sir, are you there?
Hello, everyone.
Hello.
Hello, ma'am. You are audible. Okay.
I need
So-
permission as host because I can't share the screen. Hey, এটা কি? নামাতে হবে? ও, হ্যাঁ, ঠিক।
Ma'am, are you sharing your screen?
Is it visible?
Not yet, ma'am.
Not yet. But I just shared it.
Yes, it's now visible.
Good.
Yes, ma'am.
Okay. And now I'll make it full screen then.
Yes, ma'am.
Yes.
Okay, ma'am. So we now continue with session 3, and it gives me a great pleasure to invite Dr. Nandini Chatterjee, Professor of Medicine at IPGM Kolkata, and Scientific Chairman of APA West Bengal, to speak on study design, choosing the right approach. So over to you, ma'am.
Thank you, Partho. So, a very important aspect of doing research is to design the study. Just a minute, huh? Just a minute. So today, I will be discussing on the different types of study designs which a researcher needs to have a very clear idea about before embarking on any work. We'll also be touching upon what is a qualitative study and what is a quantitative study. Then a brief overview on observational, interventional studies as well as systematic review and meta-analysis. Next, we will be dealing with the relative significance of these different types of studies, which is more evidence-based, which is having a more strength of evidence that we will be learning. And in the last part, we'll be discussing what are the different situations and what could be the ideal study design for those situations. So, what is a study design? A study design is a specific plan or protocol for conducting a study. It allows the investigator to translate the conceptual hypothesis into an operational one. So there are different types of research. They can be divided as quantitative research and qualitative research. So one by one, I will go into the different types of research. So first, what is the qualitative research and how it is different from the quantitative research? When we do quantitative research, we can quantify a problem and generate numerical data. This numerical data can be transformed into usable statistics. After the results are available, we can generalize the results from the sample to the population. An example is that if we want to estimate the burden of anemia among adolescent girls. So what we want to know that what amount, what percentage of adolescent girls are having anemia. So when we are studying that, that is a quantitative study. Another example can be that we are estimating the burden of influenza among a population of age above 65 years. So we will study those elderly people, find out if they have fever, we take their nasopharyngeal swabs and find out if they have influenza or not. So that percentage, when we do statistics with it, it will be a quantitative study. So on the other hand, what is the qualitative study? The qualitative research is exploratory research where we use it to gain an understanding of the underlying reasons, opinions of the subjects, and it provides insight into a problem. So, an example is that what are the reasons for preference of home delivery with the traditional birth attendance in rural West Bengal? So what will we do? We will make a questionnaire with open-ended questions. We will ask the birth attendants why you prefer home delivery. And different birth attendants will give different opinions. And from that, we can have some idea about the reasons behind. Of course, we cannot have numerical data in such a situation. So, qualitative research helps us to explain factors that influence health and disease, assess how individuals and communities understand health and disease, and also study human behavior and social work. This type of research can answer questions on why people behave the way they do, how opinions and attitudes are formed, how people are affected by the events that go on around them. Another example is, suppose you give a lot of questionnaires to students and ask them through way of a questionnaire that what is their perception or feedback about a particular teaching module. So in those questionnaires, the students will give their opinions, and if now a person sits and synthesizes all these answers and data, they can have some idea, a qualitative idea about the student perception. So, is this type of research enough? Well, it is not enough. So, we need another type of research, which is the quantitative research, where we can actually count and calculate the numbers and find out information about various health-related or disease-related information in the populations. So, in order to now delve deeper into this subject, we have this slide. Now, this slide is actually the crux of today's talk, where in one flow diagram, I am classifying the different types of epidemiological studies. Now, the classification begins from the point where we have to decide whether the investigator is doing some intervention or not. So, if your investigator is doing some intervention or doing some exposure to a particular intervention, if it is there, then that becomes an experimental study. If the investigator does not perform any intervention but just observes the scenario, that is called an observational study. So, in the experimental study, there will be groups of subjects who will be allocated into groups like group A, group B, and one of the groups will receive either a lifestyle modification or a new medication or some form of surgery, and his outcome, that group's outcome, will be compared with another group which is either receiving nothing or receiving something else. So this allocation into groups and comparison with the involvement of a particular modality of therapy is a controlled trial. Now this division into two groups may be randomized or non-randomized. So randomization is a process whose details will be discussed in the workshop in other talks, but where the person or the subject has equal chance of being either in group A or group B. So it depends on chance. Whereas non-randomized trials, the investigator will select the subjects. So this is the experimental study. Whereas what is the observational study? Here, as I said, the persons will be studied without any modality of therapy being introduced into the subjects. These groups, if we are just observing without comparing with another group, it becomes a descriptive study. And if there is a comparative group, we call it an analytical study. These again have further divisions, after which I will be coming as I go and progress with my talk. Now, here, as I was to continue the classification, the difference between the descriptive study and the analytical study is, in the descriptive study, we only study one group of people. At the start of the study, there is no hypothesis regarding the cause and effect, but as the study ends, we can have a possible hypothesis but cannot confirm it or reject it. Whereas in an analytical study, you have two groups that are compared, and here you begin with a definite hypothesis, compare the results between the two groups, and at the end, you can actually confirm or reject the hypothesis. So, let us have some further detailing about the different types of studies. So, descriptive studies is the first phase of epidemiological investigation, where the investigator is observing the distribution of disease or health-related event in the human population. We can also identify characteristics with which the disease is associated. Now, what are the types of descriptive studies that we know? They can be case reports, case series, and cross-sectional studies or surveys. Now, the photographs given here are of case reports. As you know, the case report is just a detailed description of a single case. So, technically, it cannot be called a proper study, but it is also a scientific observation of a particular unique presentation. The first photograph shows that ITP is happening in a patient of carcinoma tongue, which is a very rare occurrence. So this has been reported, and people who read this, they come to know that this ITP can also be a complication of a malignancy. In the second photograph, we have a common disease like dengue with an uncommon complication. So this case of dengue fever, it presents with splenic rupture. So such a reporting of a case will be a learning point for the reader, who will know that we have to be very cautious with dengue patients, and they can have this near fatal complication of splenic rupture. So, case reports are always cases who have something to teach us. Now, it may be a rare case or it may not be a rare case, but has some very important message. So generally, a new or unique finding is reported, either a previously undescribed disease or an unexpected link between diseases, an unexpected complication like the second case, or sometimes case reports can involve therapeutic adventures also, adventures or misadventures, where you can describe a new therapeutic effect of a particular drug or a very important adverse event due to a drug. Beyond case reports, there is another type of descriptive study in a very small scale, that is a case series. So, here cases or patients with similar diagnosis can be grouped together. They may be identified from a single center or multiple centers and reported as such. So, a case series a little ahead of the case report. So, more than 4 cases together, they may try to give some important learning points for the reader. So, again, here are some examples of case series that have been published in certain journals. The cross-sectional study. Now, the cross-sectional study can be descriptive or it can also be analytical. So, descriptive cross-sectional studies simply characterize the prevalence of one or more health outcomes in a specified population at a single point of time. So, the last 3 words, single point of time, is very important. It means that the investigator is evaluating or examining the patient on one occasion only. He is then taking data about various aspects of his health. He is examining him, he is taking his history, and from that he is trying to gather as much information as possible. Now, we were thinking of doing a study on people who do not have any symptoms and who are unaware about their hypertensive status. So, we had designed a study where the doctor will be taking blood pressure measurements in an accurate fashion on people who are not aware of their hypertensive status. And we will just record their pressures, take some history about comorbidities and family history, and we need to find out that how many of these unaware people ultimately get diagnosed by this method to be hypertensive. So, we are going to find out the prevalence of hypertension in people who are unaware and asymptomatic. So, this will be done by measuring blood pressure only on one occasion. So, this is going to be a descriptive cross-sectional study. This photograph, this article that was published, here the investigators did kidney biopsy. So, they did kidney biopsy in a single center and they have just simply published their reports as to what percentage of various kidney involvements have been found by doing kidney biopsy one time on such patients. So, descriptive studies, which I have just described the different types, are useful for hypothesis generation and they may also be informative about rare diseases, but they cannot study cause and effect relationships. So, let us now try to find out that if we want to investigate a causal relationship, what are the type of studies that are at our disposal? So, these type of studies are the analytical studies. Now, what are the different analytical studies? They are ecological study, cross-sectional analytical studies, case control study, and cohort study. A little detail about each of them. So what is an ecological study? Here, the ecological study study the effect of environmental exposures on different health outcomes. It looks at associations between the occurrence of disease and exposure to a known or suspected causes. However, the unit of analysis here is not an individual patient but a whole population. Now, nowadays, there is so much pollution and so much changes in the environment, in the climate. So, they definitely have effects on human health. And therefore, ecological studies are becoming very important. So, we can study a population who are exposed to certain adverse environmental factors, and we can compare them with people who are not exposed to those factors and ultimately see what is happening to them, what kind of adverse health effects are happening to them. So here was an ecological study done where they studied about environmental chemicals and which of them were having an effect on breast cancer. So they concluded that styrene was a chemical which was found mostly in a particular state of Texas, and they actually said that it was more associated with breast cancer when they compared with many other toxic materials, almost 12 of them. They studied them, they compared them, they did an analysis, and they come to the conclusion. So here is a case, not a case, but a situation or a study that was done. A defined population of school-going adolescents, and they had two groups. They were divided into two groups. One group of people or the adolescents were playing outdoor games regularly. Another group was not playing outdoor games. It was studied that what was the prevalence of obesity among the people who were having regular physical exercise and what was the prevalence among those who were not playing outdoor games, and this was compared. This was done on a single point of time. That is, they reached out to the population, they took the history of them about their physical activities, and they also measured their weights and BMI, and then they did a statistical evaluation and came to a conclusion. So, since this was a one-time affair, so this is an analytical cross-sectional study. Data was collected at a single point of time. Investigators, they collected the data of both the exposure and the outcome at one specific time for the purpose of comparison of the outcome differences between the exposed and the non-exposed subjects. So this is a pictorial representation of what I have been saying this time. So study exists at this point of time, and the study population is divided into two groups, one who has no disease and one who has the disease, and then we check out whether they have the exposure or not. Coming to case control studies. Now, case control studies, the beginning point is to start with people who have the disease. Then we match them with controls who do not have the disease. And then we have to look back. So we take the cases, we take another group for comparison, and we look back into whether they were exposed to certain factors or not. So here is an example where the cases were obese individuals, obese adolescents, and the controls who had normal BMI, and they took the history of their past activities. And from there, they would have to gain the information. So a case control study is a retrospective study where the study population is the diseased population. The diseased population is compared with the control, and then it goes back to find out whether there is any exposure or not. Such studies have been done with lung cancer patients as well. So lung cancer patients were asked about their smoking in the past, how many years they have smoked, what is the pack years, and then the causality have been found out when they were compared with people who did not have lung cancer. Next comes the cohort study. So again, the analytical studies, the case control study, the cohort study, and the analytical cross-sectional studies are the three-pronged approaches where you have comparative groups. So what is a cohort? We often hear about this word. It is defined as a group of people who share a common characteristic or experience within a defined time period. Now, the cohort study, it begins with a disease-free individual. And you divide them into groups according to whether they are exposed or not exposed. And now you keep on following them up. So you keep on following them up and record outcomes in the groups. After the outcome, you compare. So the cohort study, we have to have a number of follow-ups, and the study begins at this point of time. The study population is free of disease. Then one group, the exposure is present. In another group, the exposure is absent. And then you find out whether in future these people are developing the disease or not. So again, the same example in a different way. So the cohort of school-going adolescents with two types of exposures, and you follow them up into the future, finding out what happens to them. However, here there is one ethical question which later on may be discussed. That is, when you know that such people are not doing anything and have a risk of developing some disease in the future, then will we sit and not give them the lifestyle modification? So this is a question. This is a question, and there is an ethical query involved. So you know that people who are not having any physical exercise at a young age will become obese in the future. So you are observing them, you are following them up. So will you not give them advice that please do not do this, please start exercising? So there is this problem. Now, after the observational studies, we come to the experimental studies. So what are the experimental studies? In other words, they are the interventional studies. Now, in these studies, what happens is there is some intervention or manipulation which is deliberate by the investigator. The aim of the experimental study is to provide a scientific proof of the etiological factor and to provide a method of measuring the effectiveness and efficiency of various health services which may treat or prevent disease. So, the investigator is doing some intervention, and then we are studying whether that intervention is having a good outcome or a bad outcome, and we are always comparing it with another group who is being resorted to some other mode of intervention. So what are the different types of these experimental studies? When we do it on patients, they can be a randomized control trial or a non-randomized control trial, depending on how you have selected the patients to be put into different groups. If sometimes you perform them on communities of healthy people, you call them field trials or community trials. Randomized trials are where the candidates are selected by chance, which means that by the process of randomization, you ensure that each subject has equal chance of being in either one group or the other. Whereas in the non-randomized trials, participants are allocated into treatment and control groups by the investigator by specific inclusion and exclusion criteria. Now, the non-randomized trials require lower cost and less planning, but there may be some selection bias. And the insights may not be that valuable as the randomized control trials. So here is a picture where the study is being started. The obese students undergo this intervention. There is a control group and we are studying them in the future about the effect of that intervention. This is a publication where elderly patients with diabetes were either given oral agents or given basal insulin and their outcomes were studied after some. A community trial or a field trial is an experimental study that tests interventions on entire groups or communities, as I have already mentioned. This is usually used in public health research where interventions like vaccination programs or health education campaigns may be done in communities and they may become compared with other communities where such intervention is not being done. So that is, you call it a community trial. Now, so more or less, I have described the basic types of study designs that are useful for research. Now, if we want to find out the prevalence of a particular disease, we will resort to a cross-sectional study. But if we want to find the incidence or the occurrence of new cases of that disease, then the cross-sectional study where you will see the patient for only one time is not helpful. In that case, you have to have multiple follow-ups of the patient and go for the cohort. Similarly, if you want to find the natural history of disease, to comment on the prognosis of that disease, or even go for a causal association, you have to resort to cohort studies. The case control study is also another means of causal association evaluation. If you want to find out the effect of a particular drug, effect of a new treatment, then you have to go for randomized control trials. Like so many new drugs are coming up every year, new diabetic drugs, new obesity drugs. How are they coming? They are coming by these randomized control trials where people are being given that drug, it is being tested, how effective they are, how safe they are. Once they are found to be safe and effective, then only that drug will come into the market. So that is the main importance of the RCT, the randomized control trials. So what is the hierarchy of studies? Hierarchy means hierarchy means that there are, as I have described, there are different types of studies that are known in research. But you will see that some of the studies, their evidence strength is not much. whereas there are certain types of studies which are very much believed to be having strong evidence base. So the case report, the case series, or the cross-sectional studies, these studies we consider as letting us know about various types of disease-related conditions, but their evidence strength is not much. But when you are doing the cohort studies, the randomized trials, their evidence strength is much higher. The highest rank is occupied by the systematic reviews and meta-analysis. So what is this systematic review and meta-analysis? I will try to explain in brief. Now, if you go to the internet and type down a particular question, so you will find that thousands of studies are coming up on a particular query. So, is it humanly possible to read all those 1000 studies? It is not. So, it is a great challenge in the modern day to keep up with scientific advancement or keep up with available literature. There is so much. So, what we want, what we need is a summary of the outcomes of various trials, which you can read and have an idea about the bottom line or the gist of all these studies. So the systematic review does just that. What is that? So first I need to tell you that there are three types of reviews. Now these reviews are basically the experts are themselves reading up for you and they are trying to summarize the results or the evidences that are coming up and presenting them to you in a believable way. Now, while all meta-analysis are based on systematic reviews, not all systematic reviews necessarily include meta-analysis. I will come to this explanation of this sentence. I think some of the slides I have jumbled. Yes, some of the slides are not here. Well, the systematic review has a way of first extracting all the studies and abstracts that belong to a particular research question. So if you have a particular research question, first they will systematically conglomerate all the abstracts. There will be definite selection criteria by which they will select those abstracts. Once that list of abstracts has been retrieved and reviewed, then the process of extracting data will begin, and this will be done by two expert reviewers. Now, a simple data extraction will be done from the information from each reviewed study, and at last, these data will be synchronized to give a conclusion or the main message from all the reviewed studies. If statistical processes are used to synthesize data, then it becomes a meta-analysis. So systematic review is only the comprehensive list of the abstracts together, but statistics is not done, and if statistical data is presented or synchronized from that, it becomes a meta-analysis. So here, an individual reader did not have to go through hundreds of studies himself or herself. They are getting the gist or the main summary of the scientific information gathered and which has been done meticulously by expert reviewers in the field. So now I've almost come to the end of my talk, and here just wanting to test which is the ideal study design when you have a rare case of dermatomyositis coming to the hospital. Well, if there is only one case of dermatomyositis which has come to the hospital and you wish to document it, it becomes a case report. However, if there are 5 or 6 dermatomyositis cases coming to your hospital over a period of maybe few years, then it is important that you can write out a case series out of it. And that will give more information than the case report per se. If you want to find out the prevalence of hypertension among adolescents, this is just an example. So we will go for a cross-sectional study. If you want to find out the association of bottle feeding with the incidence of diarrhea, so you have to follow up that child for a few years. If there is history of bottle feeding and then the patient is followed up for a few years, we can see what is the incidence of diarrhea in that child, and we have to of course compare it with another child who has been breastfed. What is the association of smoking and MI? Well, if such an association needs to be found out, then it is good to start with cases of AMI and go back, take the history, whether they were smoking or not. So, people who were smoking and people who were not smoking were now compared. So, case control study where you start with the disease itself, people who were having myocardial infarction, and we go back with their association of smoking. If you want to find out the safety of a particular vaccine, it is written malaria, but it can be any vaccine. So, in that case, we have to do a randomized control trial. If you want to explore factors of non-use of contraceptives in a tribal community, in that case, we may take up a questionnaire and ask those tribal people what is their block, what is the cultural problem, what is their hesitancy in contraception, and in that case, we can have a qualitative study. If you want to find the best practice as per evidence-based medicine, there is no other way but to resort to systematic reviews and meta-analysis. So, I think I have tried to compress a number of things within this talk, and it is not easy to digest if you have not actually started to do this in your practical day-to-day practice. So, different types of study designs, if you just listen to a lecture, that is not enough for developing a concept about these things. So, once you actually get involved in some kind of study, you can start with a simple observational study. When our students are doing their dissertation or thesis, often in the thesis, people are doing those observational studies. Sometimes they do cross-sectional analytical studies. So up to that, our students, our PGTs are exposed to. But beyond that, if you have to actually understand and learn, then we need to get attached to such study groups. And our API and Physician Research Foundation has taken up a venture of involving people into various types of research, and I'm sure this workshop is the first step. Thank you.
Thank you, Dr. Chatterjee, for that lucid and well-structured presentation on research design. So as we running before the time, so we may we are ready to take questions and queries, but I think there is no question or queries in chat box. So, without wasting any time, to conclude today's academic sessions, we are joining by Dr. Indranil Shaha, scientist E at ICMR, who will speak on the critical topic of validity, bias, and confounding, essential concepts that that determine the trustworthiness of our research.
Thank you. Thank you.
So, Dr. Shah, the screen is yours.
Dr. Chaudhary, and first of all, I'm grateful to Association of Physicians of India for giving me the opportunity to talk before you. So, without wasting time, can I share my screen?
Sure, sir.
Okay. Is my screen visible? And
Yes, sir.
am I audible?
Yes, sir. Your
Okay.
screen is visible and you are audible too.
Thank you. So, good evening, all of you. So, again, I'm grateful to the organizations for giving me the opportunity to talk on this very important issue on validity, bias, and confounding. So, this is a famous quote by Abraham Lincoln. So, what he has said, if I had 60 minutes to cut down a tree, I would spend 40 minutes sharpening the axe and 20 minutes cutting it down. So what does it mean? It means that the preparatory phase, that is most important. The same is applicable in our research as well. Whenever we want to do one research or whenever we will go to some research, so it is very important that we should give enough time in the planning phase so that we can design the methodology flawless. We can choose the appropriate study design, what Professor Nandini Madam has just now told, and we can sharpen the methodology as usual and as applicable. So that we have to spend enough time in that so that the next step will be much easier for us. So what are the common pitfalls in causal research? So whenever one of our intention in the research is to see the causal association. So one is exposure is there and there is one outcome. So what is the exposure and what is the relationship between the exposure and outcome? So during this determination of the causal association, we find some pitfalls. So number one of them is the bias. So what is bias? It is a differential error in the design, conduct, or analysis of a study that results in a mistaken estimate of an exposure's effect on the risk of disease. The definition seems to be quite complicated, but the thing is this, we have conducted one study and we have seen one association between one exposure and the outcome. And we have shown that either there is a positive association or negative association. But during this showing of association, either positive or negative, we have done something wrong. That is why it has come as positive or negative. But actually, either there is no such relationship exists. So this is known as the bias. Second one important factor is the confounding. So what is confounding? So confusion of two supposedly causal variables so that part of all of the purported effect of one variable is actually caused by the other variable. So we will talk into it details. So this is known as confounding. Other two common pitfalls that we commonly see, one is the random error. So this is a non-differential error. How it differs from bias? Bias is any systematic error. So always it shows either in a positive direction or in a negative direction. In contrast, random error is either it may be on higher side or it may be on the lower side. So this is how random error differs from the bias. And the last one is the synergism. Synergism means whenever we are adding 2 plus 3, it is not coming 5, it is coming 6. So what does it mean? Relationship between exposure and outcome. If exposure 1, suppose the outcome is 2, and the exposure 2, the outcome is 3. if it acts independently, but when it acts concurrently, then it is not 3 plus 2 equals to 5, but the outcome is coming 6 or 7 like this thing. So two of them, they have interacted with each other so that the sum of the outcome is more what is expected from alone. So these are some of the common pitfalls that we measure, that we see in the causal research. So first of all, I'm talking now about the validity. What is the meaning of validity? Validity means accuracy of measurement. Suppose I have brought one sphygmomanometer blood pressure machine and I am measuring your blood pressure. Suppose I have measured your blood pressure and I have told your blood pressure is 130/90. Now from your experience, and you measure your blood pressure regularly, and from there you can say, yes sir, your measurement is correct. How do you say? Because you are quite familiar with your blood pressure and you measure it regularly. And with my new instrument, it is also giving more or less similar result. So this is known as validity, right? So again, so it refers to what the extent, what the test accurately measured, what it purports to measure, right? So there is, in the research, there are different types of validity we talk about in the research. One is we talk about the validity of the studies. Right. So whenever we talk about the research in the validity, so it might be applicable in the validity of studies. Again, it might be applicable in the validity of measures. That is, we are using different tools or instrument or questionnaire. So in that way, the validity might come. Again, sometimes we use the screening test or the diagnostic test. In that way also validity will come. So what are the things that are related with the validity of a study? So when we talk about the validity of a study, it is usually of two types. One is the external validity and second one is the internal validity. External validity means generalizability of the study. Suppose I have done one study in my own hospital among our study population, and how my findings is also applicable to the general population like this thing. This is known as the external validity. So is it clear? So external validity means how the study population whom I have selected, how they are representative of the total population, so that we can say at the end of my study that the study findings, it is not only applicable in my small swamp sample, rather it is applicable to overall population also from where it has been taken. So the findings of the study could be generalized to the broader population. So how can it be achieved? From the discussion, it is very clear that has it to be external validity, we have to select the study participants from the target population very strictly, so that it can be best achieved if I employ different random sampling technique. So if I employ different random sampling technique in selecting my study participants, then we can certainly say that my study is externally valid. So my study is also applicable. The findings of my study is also applicable to the study population. What are the different random sampling technique that we most commonly use? You know all of this. I am naming all of them. One is the simple random sampling technique, or it might be systematic random sampling technique, or it might be stratified random sampling technique, or cluster sampling technique, like this thing. So this is all about external validity. Now that comes to the internal validity. Now I have selected the study population from the target population. Now how I am conducting the study, how accurately I am conducting the study, so that the findings will be meaningful, findings will be relevant. So that is the domain of internal validity. So extent to which a study accurately measures what it intends to measure. So external validity is mostly applicable, mostly related to the sampling procedure, and it can be it can be assumed, it can be done by adopting different random sampling technique. In contrast, internal validity is the very how accurately we are conducting the study. So loss of internal validity is also called bias. We will discuss in details about the bias. Next, how we can achieve internal validity. Since I have taken how correctly, how well I'm conducting the study, it is basically the domain of internal validity. So it can be achieved by randomization, that is applicable in the randomized clinical trial, where we distribute the study population either in intervention arm or in control arm. You have heard about the matching. Matching we do in a case control study, where we select some cases, those who are having the disease, and we also select the controls who are not having that particular disease, against the adherence of the standard operative procedure, that is the adherence of the strict selection criteria, strict operational definition regarding the case definition, etc., etc. So these are all of the things that are relates to the internal validity. So if we adhere all these things, we can say we have minimized the bias. So in that way, we can say we have tried to maintain the best of the internal validity. Right. So this is all about validity of a study, external validity and internal validity. Next comes, so this is the thing you see, the small circle, it is basically taken from a large circle. So it is basically done, it is based upon decision among sampling. So if we go for the random sampling techniques, what I have already told about, so in that way, the external validity can be maintained. Now, what about validity regarding the tools? There are so many terms. I will not go into details due to paucity of time. So whenever we talk about validity of a tools, say for example, I have doing one study with one new instrument or with some new questionnaire. So in that way, we can, that is basically we are talking about the tools. So for that particular tool, what are the different ways of ascertaining validity? One of the two things are there that is based on a judgment. This is known as phase validity, and second one is known as the content validity. Suppose I have prepared one questionnaire to find out the anxiety level of some adolescent age group. So I have prepared one questionnaire. Now I am distributing the questionnaire to some of the experts. The experts will see the content of the questionnaire, and on that basis, they will say, yes, the questionnaire is good or questionnaire is bad, or you edit the questionnaire accordingly. So this is known as the phase validity. It is the weak form of validity. So second one is the content validity. It is more or less similar with the phase validity, but in spite of subjective assessment, there is also having option one, objective assessment. In content validity, we give some Likert type of scale and we ask the experts and you mark accordingly. And with that marking, we calculate one score that is known as the content validity ratio. So content validity is slightly superior compared to the phase validity, where some objective parameter is obtained, and on that basis we can say whether that new questionnaire or the new tools, how much it is valid, how much it is appropriate for that particular condition. Again, this is about the based on judgment. Next, we can also judge the tools according to the checks against some data. So these are different terminologies that is being used. I think you have all come across all of these terminologies. One is known as the criterion validity. Second one, convergent discriminant validity, construct validity, predictive validity, and the responsiveness. I'm just telling about you regarding the criterion validity. What is criterion validity? In criterion validity, we do one thing. Suppose I have developed one new questionnaire to see anxiety among the adolescent population. And say, for example, in adolescent population, to see anxiety, there is already a gold standard question. So in criterion validity, what we see, I will measure the anxiety of the adolescent with my new questionnaire. And at the same time, I will measure the same adolescent population with the gold standard questionnaire. So now we will get two results from the same population. With the gold standard questionnaire, I am having one finding. And with my new questionnaire also, I am also having one finding. Now I will match the two findings to see how much my new questionnaire is valid or appropriate in comparison to the gold standard. So these are some of the other terminologies: convergent, discriminant, constructed, predictive, responsiveness. I'm not going into details. So these are all about to see the appraising validity when we are discussing about the tools. Next come about, suppose we have introduced some screening test or we have introduced some new diagnostic tests. Now I want to see the validity of the test. Again, you see, with the gold standard, you have to compare the newly designed screening test or the diagnostic test. And for that, what parameter we have? We have one sensitivity and we have one specificity. So these are the two measures that are being commonly used to judge the validity of a screening test or the diagnostic test. So these are all about validity of the different studies. Next comes to the bias in research. Whenever we talk about bias, you will come across so many types of bias. But so this is any systematic error. I have told you in the determination of the association between exposure and disease. So we have measured the association between exposure and disease, but there is something wrong in between. So that my result is either inflated or my result is weakened. So bias compromises internal validity of the research. I have already talked about the external validity and the internal validity. So bias hampers the internal validity of a research. Again, when bias can be introduced in a study, please remember, bias can be introduced at any stage of a research study. Now, I will show you one picture. From that picture, you will see that where bias can come and where bias can distort your findings. So this is the one of the picture that I've taken from one of the article. So major sources of bias in clinical research. You see at the bottom of the slide, you see the trial progression. So trial planning, trial implementation, and last stage is the data analysis or publication. So before you have started the actual trial, that is you are in a planning stage. So from that also there might be the occurrence of some biases like flawed study design. What I was telling at the very beginning. So it starts the pre-planning is very, very crucial. That is why I've told you the quote of Abraham Lincoln. So 40 minutes in designing and 20 minutes in cutting the tree. So flawed study design, selection bias, channeling bias. So these are some of the biases that might affect the internal validity of the study, and that might quite be assured if we take care of all these studies, all these things in the trial planning. So in the planning stage, we can take care of all these things so that the bias will not be introduced in my study. So that is all about the planning stages. Now planning is over. I'm starting my trial. I'm starting my design. So in that phases, there are so many types of bias that might come up in the study. So one of the common of them is the interviewer bias. Another common bias is the recall bias. Another common bias is the misclassification or exposure or outcome. So I've written so many things, but you remember these are the major biases that might come up. So interviewer bias. So the interviewer who is doing the interview, he or she is introducing some error, or recall bias is there. So I'm asking the participants regarding one exposure and he or she is thinking and accordingly she or he is telling the information. So in that way, that might some wrong information might come up and we are noting it in my case history form. And last, another one is the misclassification of the exposure or outcome. So I have not correctly identified it as an exposure or not as an exposure. So this is my fault actually, right? So these are the different types of bias that might come up during the trial implementation stage. And at the last stage, when your trial is over, you are doing the data analysis and you want to publish the trial in a high impact factor journal. So in that time also some biases might come. This is known as the citation bias, and again one confounding that might come. So confounding, I will talk about. Citation bias means whatever similar type of studies that I'm citing and what are the studies that have come up that is against my findings that I'm not citing it in my report. So these are some of the common biases that we commonly face during conduction of a trial or during conduction of a study. Starts from the planning phase, implementation phase, and the data analysis or the publication phase. Broadly, bias is classified into two types. One is the selection bias and second one is the information bias. So what is selection bias? From the name, it is implied that we have done something wrong in selecting the study participants. Please remember, it is not related to the external validity. This bias is only talking about the internal validity. So selection bias means we have correctly identified the participants by random sampling technique into my study. But in my study, I have wrongly assigned them in intervention or control arm. So that is my fault. So selection bias might be error in selecting study groups. So it might result from this one, or it might result from self-selection bias. Sometimes you see some advertisement is given. It is also common in some foreign countries. Suppose one student from India, he is visiting one abroad country, and he has seen one advertisement. Okay, you are welcome to participate in a study from where we will ask you some questions and we will take some blood parameters, and accordingly we will see something like this thing, and for that you will get some remuneration like this thing. So now from one Indian student, he has got abroad and for a study. And again, when we see he or she sees that one, and she is again registering himself or herself in the study. But again, you see the study population, it is in the foreign, so that socio-demographic condition that is not according to the parent population. So in that way also some bias might be introduced if he or she is registered in the study. Again, another important study was there in one of the industry. The industry was mostly working with the formaldehyde, and some authorities were telling that you are using formaldehyde and you are not giving any protective instrument to the workers, so that the workers, they are facing more eye irritation and eye problem. So to confirm this, the one industry, they conducted one study, and they have shown that you see the workers, most of the workers, they are not facing such problem. But what the problem is like this thing, most of the workers, those who face the problem, they leave the industry. Right? Suppose initially there are 100 workers, and with passage of time, 50 or 40 workers, they are facing problem, then they left the company, and remaining 40 people, they were not facing any problem, and only those 40 people, they were included in the study. Now I'm doing interpretation. Yes, I've studied 40 people, and there is absolutely no problem with the formaldehyde and the eye problem, despite they are not using any protective device. So in that way also, this is also known as the self-selection bias. Again, another bias that might come up, that is known as the non-response bias. In one of the famous study, it was shown that allergen and some dust allergen and some chemicals, they were mostly responsible in causing the allergic rhinitis, asthma, like this thing. And they have conducted the study, and they showed the response rate was 70%. And they have shown that there is no such positive relationship between the allergen and the causation of asthma and allergic rhinitis. Then one scientist, he became interested to see, and then he saw that 30% they did not respond, and he went to those 30% and he interviewed them, and then he saw that non-response who did not respond and who were not included in the study, they were quite different from those who participated in the study. So in that way also bias has incorporated in that study. So these are different types of selection bias. Now you see another type of bias is known as the information bias. What is information bias? It might result from recall bias. I am asking one participant, the participant is not able to tell when he or she started smoking and what was the frequency of smoking at the initial stage. Right, so in that way, some recall bias might come up. Again, another important thing is this: it has been seen that a study between the defective birth, right? So after the birth, there is a live baby, and the baby, there was some development, some problem was there. And then with the problematic child, the researcher, they asked the mothers, and the mothers who gave born the defective child, they were more critical, and they were telling each and everything. Okay, I ate that chili powder, probably that might be the reason like this thing. So it is actually not the case. So in that way also, since it is based upon the responses from the participants, in that way also the recall bias, it might come in the information thing. Another one is the interviewer bias. Suppose I am interviewing the participants, and from the beginning I know which participants they belong to the intervention arm and which participants they belong to the control arm. Right? And intervention arm, they are getting the new intervention. So in that way, I'm asking them, okay, in the intervention arm, I think you are feeling very good. No, now your problem, headache, reeling of head, these are all very less. So in that way, you see, I'm just judging the participants to tell what I intend to listen from them. So this is known as the interviewer bias. Another type of bias that might come up, this is known as the misclassification bias. Suppose I did not measure it correctly. Suppose I have taken a case control study. I've taken lung cancer patient and non-lung cancer patient. Again, lung cancer patient, how have I judged? I have judged from the history and the chest X-ray. Suppose a very new lung cancer, it might not be detected by chest X-ray. So I did not do any CT scan of the chest, right? So in that way, some of the lung cancer patient I have mistakenly taken into the control group. So in that way, I have incorporated some error in the classification. So this is known as the misclassification bias. So grossly, these are the different types of biases that are commonly seen in the research study. Now the question is how to mitigate these things. So how to handle this? So these are the handling procedure. Error in selecting study groups, we can do it if we correctly do its randomization and we correctly do the matching. Again, randomization is only applicable in the randomized control trial. Matching is only applicable in the case control study. Again, other than this, if we adhere the selection criteria, then we might solve some of the problem. Again, during the analysis phase, there are some statistical application like standardization, like multivariate analysis. So with this process, we can take care of the some sorts problem of the selection bias. Again, the non-response bias, I have told you, you have to separately, you have to do analysis of non-responders. You cannot access all the non-responders. Some of the non-responders you can access, and from that, if you take the data, from that you can have a broader picture whether the non-responders, they differ systematically from the responders or not. These are some of the mitigation theories that you can take care to address the selection bias. Regarding the information bias, to address recall bias, you see, you can take some objective data source as well. So in that way, and you can take care regarding this thing. So in that way, you can take care of the recall bias. Interviewer bias, I have told you, I knew in which grade group they belong. So you make me blind. So double blinding will take care of the interviewer bias. Again, misclassification bias, you see, you take the standardized procedure, standardized gold standard procedure. You don't take lung cancer and non-lung cancer patient based upon a chest X-ray. You go for a higher version from where you can accurately diagnose. So standardized procedure, again, you have to very much adhere with the operational definition. When we will say that it is a case and that is a disease positive, and when it is not a case, that is a disease negative. Again, if you are not very sure, you can do a pilot study as well. That is, you run the research in a small scale, so that in that way you can identify the different types of biases, and then you can have an idea how to solve the problem. So these are all about different types of biases and different types of solution to address each of the biases. Now the last part is the confounding. Now this is one of the famous study in the history. So Best and Walker, two scientists, they studied the mortality in 3 groups of men. So they took one group where the one group they comprised of the non-smoking population. So one group, they are entirely non-smoking population. They took the second group, all of them, they are the cigarette smokers. So all of them are the smokers. And they took the third group. In the third group, they took either the pipe smokers or the or the cigarettes, right? So these are the three groups. So one group comprised of the non-smokers, one group comprised of the cigarette smokers, and the third group that comprise either the pipe smoker or the cigarette, right? And in that three groups, they have calculated the mortality rate. So what they found in the non-smoking group, they found the mortality rate, that is the death rate, 20.2 deaths per 1,000 population per year. So in a year, there were per 1,000 population, there is 20.2 deaths in the non-smoking group, 20.5 deaths in the cigarette smoking groups. And you see 35.5 deaths in the third group, that is those who are taking cigar and the pipes. So what is your interpretation? What is your interpretation? Interpretation is like this one, that cigarette smoking and non-smoking group, the death rate is more or less same. So cigarette smoking is not so harmful. So, but you see in contrast, cigar and the pipe smoking, that is the mortality rate is quite high. So cigarette smoking, cigar and the pipe smoking, these are absolutely bad. But cigarette smoking is quite good because the mortality rate is quite similar with the non-smoking group. Then one scientist take questions about your study pop. They said, yes, all of them were men. Then he wanted to know, okay, tell me regarding the mean age, average age of these groups. Then the scientist revealed the average group of the non-smoking, they are 54.9 years. That is around about 55 years. So the average age of the men, those who belong to the non-smoking group, they were around 55 years. And what about cigarette smoking group? Now you see, they are around 50 years, 5 years younger compared to the non-smoking group. And what about the mean age of the third group? Then they saw, oh my God, it is 65.9, so much higher. So what they did, they said, okay, what is your previous figure? What you have shown, we do not believe in this thing, because your cigarette smoking group is quite younger group, non-smoking group is quite older, and again the last group is much older. So naturally we know that with age there is increase in the cancer risk. So what they did in each of the group, they stratified each of the groups. So then they did then the similar age groups of the three groups, then they compared. Say for example, those who are near 50 in the non-smoking, smoking, and the cigar group, they compared. Again, the persons, those who are mean age around 60 in the non-smoking, cigarette smoking, and the cigar group, they compared. In that way, they have calculated, and in that way they have calculated one single value. This is known as the standardized death rate. Now you see the standardized death rate of the non-smoking group, it has come 20.3, but in the cigarette smoking group, now you see the value. It has come up 28.3, and in the cigar group, it has come 21.2. You see just the finding, it has been just toppled up. So when the scientists calculated the adjusted death rate, that is they took the age. So what happened in this case? This is a classical case of confounding. The confounding in Latin roots means to pour together. So what we observe, there is one exposure and there is one disease. So when do we say a confounding factor? Confounding factor is such a factor when it is associated with both. Please remember, this is very important. When it is associated with both with the exposure and the disease. Both with the exposure and the disease. So what happens in this case? What we observe cigarette smoking is causing more death. But what actually happened in that case? Cigarette smoking causes increased death. It is true. But at the same time, it is also true that the cigarette smoking group, the age group was much higher. So age is a confounding factor here, because with increase in age, there is increase in cigarette smoking, and there is increase in age, there is increase in the death rate. So age is a classical example of a confounding factor, since it is associated with the exposure, that is cigarette smoking, and with the outcome, that is the death. So there are different types of confounder. In this example, what I have told you, this is a case of positive confounder when a third variable produces an association that actually does not exist or exists. Right. So it is a positive confounder. Again, there might be negative confounder when a third variable masks or weakens a true association between two variables. Suppose I am interested to see whether increase in exercise in the older age group that reduces the risk of heart attack. So this is my question. So exposure is physical activity, whether it decreases the occurrence of heart disease. So here age is a negative confounder. Why? Because with increase in age, there might be decrease in the physical activity. Right. Again, with the increase in age, there is increased occurrence of the heart disease. So age is in between what is right. So this is an example of a negative confounder. Again, there might be another type of confounder. This is known as the qualitative confounder. Qualitative confounder means when a third variable causes the reversal of direction of effect. Suppose one new anti-cancer drug has been introduced in a treatment of stomach carcinoma, and we have some patients and we have used that new drug against the stomach carcinoma, and it has been seen in the follow-up longitudinal study after 5 years that the survival rate is much good in among the women compared to the men. So again, here there might be so many types of confounding factor, like gender is example of confounding factor. So it might be seen that the female patient, they might adhere the treatment guideline more accurately. Number one, again, there might be some genetic association with the treatment or with that particular drug and the stomach cancer. So in that way, the qualitative confounder might come out. So please remember, confounding is a factor that is associated both with exposure and the disease. So how to identify the confounder? So you have to identify the confounder before you have initiated the study. That is why I was telling about the planning phase. So list all the possible confounding factor on the outcome. How to make sure that it is the confounding. You can see the previous research. You can take your clinical insight. And if you have a good clarity about the pathogenesis phase of the disease, then you can identify, yes, these are the factors that might be act as an confounder, right? So I'm not telling this one. So so there is one difference between the confounders and the covariate. So confounders, those who are associated both with the exposure and the disease, but covariate, it is not necessarily associated with the exposure. So it is only associated with the outcome, right? So these are the two factors between the confounders and the covariate. So these are some of the potential confounders example. One is the age that I have already told about. Second one is the gender. Third one is the socioeconomic status. How socioeconomic status can be found as a confounder. I am giving you one example. Suppose it has been seen that suppose my research question is whether physical activity improves academic excellence. Whether physical activity improves academic excellence. So in that way, the socioeconomic status can be act as an confounder. Why? Because with increase in socioeconomic status, there is more scope, more this thing to involvement of the physical activity. Again, with increase in socioeconomic status, there is more scope of higher educational opportunities. So in that way, we are telling yes, those who are doing physical activity, they are scoring good marks. But it is not actually the case. The actually case is the socioeconomic status. It is associated with the physical activity because with more physical activity, they are they are having more access to the physical exercise, and they are similarly more access to the diverse educational opportunities. Right. So in that way, socioeconomic status is also a confounding factor. Similarly, living condition as well is also can also act as a confounding factor. So there's a high possibility that researcher may not be able. So if it like this thing to identify confounders prior to the study, so he has to adjust the confounder in the analysis. So how to handle the confounder, how to control this thing. So confounders can be controlled in two ways. We can Again, I've told you, please understand the difference between a covariate and the confounder. These are not synonymous. Again, you see, this is an example of a covariate. If my research question is, does a new drug reduce blood pressure? Right. So again, age might be a covariate because older age may influence the blood pressure as well. So including age as a covariate that will help to control for these effects and give more accurate estimate of the drug effect. So whether a new drug, whether they are reducing the blood pressure in the younger age group, middle age group, and the older age group. So in that way, the age might be considered as a covariate in this example. So how to eliminate the confounding factors? It can be eliminated during the study process. How? If you are doing case control study, it can be done by the matching. If you are doing clinical trial or randomized control trial, you are doing randomization, so the problem is solved. So in most of the RCTs and the case control studies, we can eliminate confounding factor during the planning phases, right? But if it is not addressed during the planning phase, in some other study designs, other than RCT, other than case control studies, then these are the two options. One is you have to do the stratified analysis and you have to do the multivariate analysis. Multivariate analysis, I think it will be discussed tomorrow. Different types of multivariate analysis like correlation analysis, regression analysis are there. So by that way, you can take care of this thing. So these are the two ways to control confounding during the analysis, that is the stratification and the multivariate analysis. So this is my last slide. I know all of you are very much tired. This is the last session. So what we have discussed for the last 45 minutes, we have discussed about the common pitfalls in causal research. We have talked about bias. We have talked about confounding. We have talked about random error, and we have talked about the synergism. Then we have discussed about the validity. We have talked about the validity regarding to study. One is the external validity that here can be taken care of the random process selection process, and also the internal validity. Then we have talked about the validity of the tools or instruments like phase validity, content validity, criterion validity. Next, we have discussed about the validity of a screening test and diagnostic test by means of sensitivity and specificity. Then we discussed about the bias. We have talked about two broad types of bias. One is selection bias, one is the information bias. We have talked about selection bias regarding the error in selection of the study participants, regarding self-selection bias, and regarding non-response bias. In information bias, we have talked about recall bias. We have talked about interviewer bias. We have talked about misclassification bias. And accordingly, I have also given you the solution how to mitigate the different types of selection bias and the information bias. Lastly, we have discussed about the confounding. It is a factor which is associated both with the exposure and the outcome. We have given you one example, how to confirm it from your previous literature. And I have also given you the example like age, gender, religion, socioeconomic status, living condition. These are some of the common confounders that we commonly see. How to mitigate the problem, how to solve this problem, we can take care regarding the planning process, but it is only applicable in case of case control study or the randomized control trial when we are doing matching and randomization respectively. But other than these two types of studies, there are also two options during the analysis phase. We have to go for the standardized analysis, that is the adjusted analysis. What we talked about in best and summer in the three groups. The last one is the multivariate analysis. So this is from my Institute of ICMR, National Institute for Research in Bacterial Infections. So thank you very much for your patience hearing. So I'll be happy if you have any questions to ask. Over to the organizer.
Thank you, Dr. Shah sir, for unpacking such complex concepts with clarity and precision. I think there is no questions or queries in chat box. So thank you, Dr. Shahasar again. And that brings us to the end of day one of this workshop. And thank you to all the speakers, participants, and organizers for making today a grand success. And we hope today's discussion have enhanced our understanding of the essential foundations of medical research. So we will reconvene tomorrow, June 26th at 3 p.m. sharp for day two, which will focus on sampling, tools, data analysis, and protocol development. So till then, take care and see you all tomorrow. Thank you. Thanks.
Thank you.
Thank you, Dr. Datta. Thank you, madam. Thank you. Good night.
Thanks.
Thank you so much, everyone, for joining. The session was very informative. So with all your permission, I'm closing the session for today. Thank you so much.
Sure, ma'am.